Good morning, good afternoon, everyone.
Good morning, Dr. Rishi, and good
morning. Good morning.
Very, very good morning.
morning, Dr. Purnima Sharma ji. Good morning, everyone.
Very, very good morning, Dr. Ravi.
Good morning, everyone.
Morning. Morning, Dr. Kanchet. How are you?
Yeah, fine. Long time
Nice
no
to
see.
Yeah, Swadee Khap.
Swadee krap, swadee krap.
Okay. Dr. Purnima Sharma ji, first time I'm seeing you very well in the webinar, like neither in a vehicle nor with any issues going on. You feel so relaxed. Morning.
Last time also it was fine, sir.
So,
Good morning, sir. Good day.
we've
Morning,
become
how are you?
wiser now. Become
Okay.
wiser. Yeah.
Right. I'm very happy to see you here.
Thank you. Thank you. Thank you.
Great.
So we'll start in 2 minutes. Just wait for 2 minutes.
कैसे गए आप?
Good morning, Dr. Patle, sir.
Good morning. Good morning. How are you? Good morning.
Fine,
Good morning,
sir.
all.
Morning. Yeah, good morning. Morning.
Just to inform that Dr. Patle is the Additional Commissioner in Horticulture in Ministry of Agriculture. And Dr. Khetrapal is Executive Secretary of APARI.
Pleasure meeting you, Dr. Naveen Patle ji.
And
Thank
Dr.
you,
Rishi
thank you.
Jagi is the key person, the coordinator of EPCOP for this program. All our speakers have joined. Dr. Jhao is not there. This is 10 a.m. I think this will start now. So good morning, good day, namaskar to all the eminent personality and all the participants. Myself, Dr. Srivikam Shukla, convener of this webinar series. I extend a warm greeting to all the participants on behalf of Biotech Consortium India Limited PCIL, New Delhi, India. To start with, once again, I express my sincere gratitude to the Asia Pacific Association of Agricultural Research Institute, APARI, and Asia Pacific Consortium of Agriculture Biotechnology, EPCOP, for giving this important opportunity to BCIL. Response of participants for all the webinars were highly encouraging. And for this webinar also, we have got an excellent response from more than 45 countries participant there with us with this program. For this series, this is the fourth and final series. Earlier we did program for the banana root and tuber, cash crop, woody species. And now this program is focused on the ornamental plants and flowering plant, tissue culture and the quality management practices. So without taking any more time, now I directly invite Dr. Purnima Sharma, Managing Director of Biotech Consortium India Limited, BCIL, for delivering the welcome address. Just briefly introduce Dr. Purnima Sharma. She is the Managing Director of BCIL, as I told, and she is the doctorate in experimental medicine from PGIMF Chandigarh, and she has been leading the many prestigious projects of BCIL, including biotech park, technology transfer, various kind of key ongoing activities of various ministries. So just a brief introduction for Dr. Purnima Sharma, and she is leading this BCIL key organization for last 2 decades. So welcome, ma'am, and it is over to you.
Thank you so much, Dr. Shukla. So before I start, of course, at the outset, I would also like to express our heartfelt gratitude to APARI, particularly Dr. Ravi Khetrapal and Dr. Rishi Tyagi, for having partnered with BCL for organizing the series of 4 webinars covering a wide range of crops aimed at popularizing plant tissue culture raised plants and for widespread adoption among the potential stakeholders, especially in Asia Pacific and the Africa region. So formally start with the welcome notes. Dr. Naveen Kumar Patle, Additional Commissioner Horticulture, Ministry of Agriculture and Farmers Welfare, Government of India. Dr. Ravi Khetrapal, Executive Secretary of Asia Pacific Association of Agriculture Research Institution, APARI. Dr. Rishi Tyagi, Coordinator of the EPCOP under the APARI. Esteemed speakers and panelists of the webinar, my colleague who's also the convener of these series of webinar, Dr. Shivkant Shukla, DGM BCIL, and distinguished participants. It gives me a great pleasure to welcome you all to this webinar on tissue culture of ornamental plants, organized jointly by BCIL and APARI. Just a little introduction about BCIL. As many of you all are aware, BCIL is an organization established way back in the year 1990 by the Department of Biotechnology, Ministry of Science and Technology, Government of India, and the All India Financial Institutions for facilitating commercialization of biotechnology by providing value-added business support services, which includes consultancy, technology transfer, project management, in various thrust areas of biotechnology, and plant tissue culture has been one of our major thrust areas right from our inception. And we were also instrumental in establishing and managing the national certification system for plant tissue cultures for almost 15 years. And that has lent us extensive experience and expertise to take forward this particular area. And therefore, we're very grateful that we would partner with APARI for organizing these series of workshops. So we are, this is the 4th, as Dr. Shukla said, this is the 4th and the last webinar in the series of these 4 webinars. And this is going to focus on the significance of tissue culture of the ornamental plants. And ornamental plants, as we know, are widely propagated through traditional methods. Of course, in some, lately, of course, it is also being propagated by plant tissue culture, which again offers a lot of advantages in terms of rapid propagation and selection of particular breeding varieties and also preservation, etc. And this is commercially also being done in India and many countries. However, quality practices is something which really needs to be strengthened. This is to ensure that we have disease-free planting material. And in this context, today's webinar, which is focusing on production of quality and certified tissue culture material, assumes a lot of significance. We are very fortunate that Dr. Ravi Kumar Patleji, Additional Commissioner, Ministry of Agriculture and Farmers Welfare, Government of India, has kindly agreed to chair this webinar. Dr. Patle is highly experienced in expertise. He's also handling the additional charge of the Director of the Central Institute of Horticulture, Nagaland, and Executive Director of the National Bee Board. Dr. Patle has more than 20 years of experience in agroforestry and horticulture research, extension, plantation management, clonal forestry, capacity building, just to name a few. He has published a number of research papers in national and international journals, 2 books and many chapters, I think more than 30 chapters. and is also the recipient of the Best Krishi Vigyan Kendra Award by the Indian Council of Agriculture Research. I extend a very gracious welcome to you, sir. We are privileged to have with us Dr. Ravi Khetrapal, Executive Secretary, APARI. And we are also very grateful to Dr. Ishi Tyagi because he's very busy. Last time, of course, we couldn't have him for the webinar since he was traveling. So he's also kindly agreed to give opening remarks covering the role of APARI in sustainable agriculture and future endeavors in the Asia Pacific region. This is going to be very, very interesting. I'm looking forward to this. And Dr. Khetrapal, as we know, has extensive expertise in the areas including R&D, policy issues, capacity buildings in sectors such as biosecurity, biosafety, and biodiversity. I extend to Dr. Khetrapal a very, very heartfelt welcome. We are delighted to have with us Dr. Rishi Kumar Tyagi, who is coordinator of the APCOP under APARI. Dr. Tyagi also has extensive experience of more than 3 decades in managing plant genetic resources by virtue of heading the Department of Germplasm Conservation at NBPGR in his earlier avatar. And I extend to him a very warm welcome. We are fortunate to have a number of eminent experts who have kindly accepted our invitation to share their wisdom and experiences by making presentations and also participating in the panel discussions and also sharing their success stories, which again is something we all look forward to. Among them, I just take names. Of course, we're looking forward to all their presentations. Dr. Thamasiri, Professor of Mahidol University, Thailand. Dr. Ashwath, Principal Scientist of the Indian Institute of Horticulture Research. Mr. Zhao, Director and President of the Grand Biotechnology Company, Taiwan. Mr. Kishore Rajhans, Director, KF Bioplants Private Limited, who is actually one of the best tissue culture companies in India, and my colleague and convener of these series of workshops, Dr. Shivkant Shukla, Deputy General Manager, BCL. Extend a very grateful welcome to all our speakers and experts. We are heartened by excellent response from the tissue culture industry, academia, research institutions, nationally, internationally, who've joined us both online and also through the YouTube channel. We accord a wholehearted welcome to one and all. Once again, a warm welcome to one and all, and thank you very much for having, included me also for presenting the welcome address. Thank you once again very, very much. Thanks.
Thank you, ma'am, for this encouraging word and setting the tone for this webinar. Now, I would request Dr. Ravi Khetrapal, Executive Secretary, APARI, kindly provide some insight on the role of APARI in sustainable agriculture and the future endeavor in Asia Pacific region. Dr. Khetrapal, his introduction is well known to all the agri stakeholders globally. He's the head of APARI, and also before APARI, he served as KB, as regional director. And he has been visiting across the globe, various countries, regarding policy matter, capacity building program, published number of more than 100 research papers, many books, chapter, contributed significantly on a very intellectual forum. So I welcome Dr. Khetrapal, sir, and over to you.
Thank you, Dr. Shivkant Shukla. Very good morning to all of you. At the outset, let me welcome again, Dr. Purnima Sharma has done quite well, but I'm sorry for my repetition. Let me welcome Dr. Naveen Patle, who is the Additional Commissioner of Horticulture in Ministry of Agriculture and Farmers Welfare in Government of India. Let me also welcome Dr. Purnima Sharma, who had been welcoming each and every one, who is a very dynamic Managing Director of BCIL. And you all know BCIL, which has a global outreach now, working on not only agriculture, but healthcare, environment, and process industry, and so on, and have their own approach to work. And distinguished speakers of the day, who is going to enlighten us on the journey from research to commercialization and on the success stories of the private sector. Dr. Shivkant Shukla, Deputy General Manager, a very active man. And I think when I look at his face, I don't look at him, I look at tissue culture. He symbolizes tissue culture in India nowadays. And my very dear colleague, Dr. Rishi Tyagi, who has been steering our biotechnology and bioresource program, which is courtesy Council of Agriculture, Taiwan mainly. And to all the participants and other who are helping to organize this webinar. Now, Dr. Purnima Sharma gave a background of like this being the fourth of the webinar series, which we had envisaged when we initially jointly collaborated with BCIL. So I may like to skip on that, and I will directly go on the role of APARI, so that we get a glimpse of at what platform we are and what we are actually envisaging to do in tissue culture and above and beyond. So APARI is a membership-based organization. We were born the year BCIL was born, in 1990, out of the womb of FAO. Like BCIL came from the womb of Department of Biotechnology in India. And we were with FAO till 3 years back. Now FAO has become our, we are dissociated, or in a way it has become like a stepmother. And we are trying to have our own legal status as a quasi-IGO in Bangkok. This legal status is very important. Once we are declared as an IGO, intergovernmental organization, the profile will change for APARI again. And it's a question of about 6 months. So with this background, I may like to say our vision is given on my background. Sorry, it's not written here. It was in the earlier version. It is strengthening agri-food research and innovation system in Asia and Pacific for sustainable development. Now we have a clear strategic plan. And in the strategic plan, we have 4 thematic areas. Number 1 is mainly on natural resource management, which include biotechnology and bioresources, under which this program is coming. Number 2 is on risk mitigation. In risk mitigation, we are working on many projects. I will come to it briefly and the importance of it. And then we have another thematic area of policy and advocacy, anything related to agriculture. And the 4th is, which is actually the development work called inclusive development, where we try to look for pro-poor value chain, which value chain everyone talks about. Now, we work with global projects from Council of Agriculture. This AppCorp is one of the important projects for us, sponsored by Council of Agriculture, Taiwan. Then we work a lot with WTO in Geneva on doing the projects on facilitation of international trade by compliances to the WTO standards through technical means, where we really need to comply with the international standards of phytosanitary measures of IPPC and of Codex. Both are important for pest or plant health and food safety. Then we work a lot with FAO Rome on the space of innovation. And not only FAO, within FAO, we have a tropical agriculture platform with which we work, which is across the world. And I was lucky to be a chair of tropical agriculture platform till 2 months back. My term is over. But what we have done there and what we are still doing is developing of an agriculture innovation system. And we have also develop the guidelines of how we can have a good agriculture innovation system, which is a big subject. I'm not going to deal on it, but which actually calls for a multi-stakeholder platform where you have all the actors, all the possible actors to come on one place and then see what can be done jointly or bilaterally. So innovation is very, very important for Pari. And here, when we talk of innovation, everyone is talking of innovation. But globally, people are now talking more of innovation than research. However, I think we are one of the very few in the world who is talking about capacity to innovate. People are not building capacity to innovate, they are building only technical capacities. Technical capacities, now what we are doing in our projects is trying to blend the technical capacity with the functional capacity. And the functional capacities are the one which leads to the capacity to innovate. This is something very unique. And after this came up, we have blended this functional capacities, the capacity to innovate, in our WTO projects and in our FAO project, and also in our project with USDA and also with European Union project, which is on the food safety ecosystem. Sorry, just one second. Okay, so we also work very closely with Forum of Agriculture Research of Africa. I mean, it's more on the knowledge sharing basis right now, but they are members of APARI. And also with the Global Forum of Agriculture Research and Innovation, where I'm happy to say I became the chair recently, which actually has more than 650 members globally. And there, what we are trying to do is, it is a network of networks. APARI is one of the networks. It's all the global network are finally with GFAR. And here, what we are trying to do is trying to bring farmers at the center of innovation, where we, right from the time we design a project up to we carry the outputs of the project in the field. So it's a slightly different concept, focus on a very innovative approach. And what we do in the GFAR is a kind of collective action. We call it collective action is the thematic area of GFAR. And here we are right now, APARI is leading one collective action in Asia and Pacific on forgotten food, and which can be in a way termed as underutilized species and all. But this forgotten food has been, it has captured the attention in the UN Food System Summit last year, where a lot of trust has been given to ensure that with all the other problems and all, we may also try to look at this part collectively, globally. So working with GFAR, with TAP, we are trying to have a good linkage globally, good concept globally. At the same time, we work now, there are 11 CGIR institutions which are left in the process of integration of CG. Now we have one CGIR, and happy to say we have been taken into the CG System Council as a member, active observer, by virtue of being GFAR chair, and it gives a very good partnership with CGIR globally. And in the region, we are working with CGIR regional Southeast Asia. It is represented by IRI, the regional director. We are working with them to have a partnership with Association of Southeast Asian Nation, ASEAN, we call. So we had a couple of meetings, and recently in Manila, when your last webinar was going on, we had finalized a kind of 8 priority areas with CG to work in Southeast Asia. Similar effort is now will be done in South Asia with the CG representative in South Asia. So we will try to see how best we can come forward to synchronize, to align, to match the areas of work, priority areas, and work collectively in the region. Then we are building the capacity in Bangladesh of the National Plant Protection Organization, a project which is finishing, a first phase is finishing this month, and next phase will start early next year, where we are trying to work with a lot of not only kind of for capacity building, but capacity building in a very holistic manner. We are not, for us, capacity building is not you train some individual and you say you have built the capacity. Our concept of capacity building is very holistic. It's a capacity building of the individual, capacity building of the institution, and capacity building of the enabling environment. And we look at capacity building in a very holistic way. I will not say much on that because I have my limited time to cover, but that actually serves much more in the long run on the capacity build. Why I'm saying so? Because individual capacity, the problem is capacity retention. You train 40 people, 20 people run away, 20 people leave the job, 20 were temporary, 20 are transferred, someone is promoted, someone is no more keen, something happened to someone. So capacity investment for individual capacity building often go waste to a great extent. It's about 70%, as they say globally, it goes waste unless you build capacity of the institution in parallel so that things move on. And that, of course, enabling environment capacity is a challenging job where you go for the policy level interventions or policy positioning. And sorry, just 1 second. Yes. So with this glimpse of what APARI is doing in terms of operations, in terms of projects, in terms of strategic interventions regionally and globally, I may like to just add, we also work a lot with SAARC Agriculture Committee. We have prepared their 10-year multi-sectoral plan, work plan, where all the 8 NARs of South Asia are there. We are right now working on their mission and vision document along with them. And we have opened our chapter with Secretariat of Pacific Community to look at the priorities of working in the Pacific Island countries. And there we had some very good meetings. Only 10 days back, I was in Canberra in a meeting of Crawford Fund, and we had a whole team of SPCE present there. So we are coming to some logical conclusion how to really work more on the ground in the Pacific Island countries, which we had not been doing much, to be honest. So, and we work a lot with the Asia Pacific Seed Association here. Also, we just got one more WTO project with them for enhancing seed trade and building a public-private partnership and to ensuring that seed supply chain is not affected globally and they follow the international norms. Now, partnership is a crucial SDG goal, very crucial, though it's the last goal in number. But I find it should have been the first goal, but it's the last 17th SDG goal. And APARI has partnered with BCIL on some very important activities, not only the webinar series, but we are also trying to develop a resource document on genome editing. And here, what our objective is, develop a document which helps us not to make the policies of individual countries, but to help them in policy positioning, give them a clarity on how their policies should look like. So we are bringing the best practices there from Japan, from Korea, from Australia, from India and Malaysia and many other places. And Dr. Rishi Tyagi is leading that thing with BCIL. And is slightly delayed, but we will ensure that this also comes up before the last quarter of the year. Now, this is a glimpse of what Apari is doing and how much we mean our business in partnership, because partnership is very crucial. The key problem is resource mobilization, funding. We are not a funding organization. We are a membership base. We have a small membership fee collector. There's no other source of income. But we get some global projects, and through project management fee, admin fee, and all, we try to do more work, and we try to give a bit to the partners who come forward to help us in our endeavor and who are very performing partners. About tissue culture, I'm not a specialist. I may not say much, and Dr. Purnima Sharma, Dr. Shivkant Shukla have said all about the importance. Dr. Rishi Kari will be concluding the webinar, so he will really highlight what is going to be there in future. But since I may not be there after 10 minutes, because we have our executive management team, our governing board meeting on Monday and Tuesday, we are just engrossed in certain formalities to be done. But I'm very happy to participate now. And I may like to leave with all of you some key takeaway for the participants, which I believe is important. One is updated knowledge on where we are with crop-specific tissue culture and its challenges and opportunities to expert speakers and panelists we have had, we are going to have. This is fine. But please see the following opportunities. Number one, areas of collaboration in research and development and innovation. Areas of collaboration through projects for promoting tissue culture entrepreneurship. This is something I believe should be one of the important outputs. A lot of research and development has come up over the years in the field of tissue culture. We need to be mindful of the fact that it is an era of innovation, which I said in the beginning also. And my request to all of you is to ensure that you capture innovation. You capture the innovations in the area of tissue culture and promote a multi-stakeholder working environment, which actually is going to be a way forward. Think of capacity building in a holistic manner. We always start with individual capacity building, but think it more in a holistic manner, bringing institutional capacity also into picture, though enabling environment capacity is a bit tougher job, and which is often taken care by the senior management. And if possible, try to blend the technical capacities with the functional capacity so that you bring some capacity to innovate. If anyone is keen, Apari can really do a bit of hand-holding on it initially, but that will be very useful. So I may like to conclude by saying that for many of Apari members and those participating from non-Apari members and from Asia and Africa who wish to promote biotechnology. When I say biotechnology here, I am more talking about the tissue culture right now. BCIL is an institution or a company, as they may like to be called, or an enterprise which can be a good model to emulate. So I would like to stop here and over to you, Dr. Shukla. Thank you, one and all.
Thank you very much, sir, for giving this detailed insight about Apari. And definitely, we have noted all your suggestions, comments, and way forward. And a BCIL and as a stakeholder of Indian industry, definitely would like to participate in some of the activities which is relevant to BCIL and the Indian context. So thank you once again. With this, I'll just take 3 minutes to give the glimpse, because this is the last webinar and we have to present some glimpse. So hardly I'll take 3 minutes to present that concept, what is the role of BCIL in promoting plant culture. Then straight away we will invite our chief guest after that. So I have made just a few slides. So about BCIL, I'm not to talk anything much. Already it has been told that it is created by Department of Biotechnology. It has been playing key role in various disciplines of biotechnology, not only plant culture, for the last more than 3 decades. So we organized 4 webinars. We had a wonderful participation, more than 3,000 registration we received altogether. And in all these 4 webinars, we talk about different topics, and all the takeaway has been noted. So definitely we'd like to formulate a good program in due course. We did various programs in Indian and global context in recent past, like as our MD told that BCIL was the key agency for implementing, not only implementing, for conceiving the idea of certification and making it a successful self-sustaining program in India. So national certification system for pisciculture plant, it was managed by BCIL for 1.5 decades. Training capacity, it is the important activity of BCIL. We did for the 25 African countries, and the Ministry of External Affairs, they are keen to have such kind of program in future. Entrepreneurship development program is the key activity, and the BCIL has created a successful first generation entrepreneur in the country. We can claim that, particularly for the Northeast region. And based on this experience, BCIL has focused 5 self-sustaining programs, because nowadays for all the activity that should be self-sustaining, revenue generating, so that whatever revenue generated, this should be plowed back for the running this program. So one is the quality stock culture. Already we have tested this concept, and very soon we are going to scale it up. EDP, we are going to organize in big way with the support of stakeholder. Some utility application we have created, because we feel that this is much needed for maintaining the documentation, quality support services, and information like what program we are running right now as a webinar training program. Definitely would like to conduct this thing. So we would like to expand this program to Asia Pacific and African region with the support of APARI and our partner. So with this thing, I'd like just to conclude that BCIL has been playing key role for the promoting plant culture, and definitely it will continue its endeavor for the betterment of this sector. Thank you so much. So with this, I am just closing this thing. Thank you so much. So now, I request our chief guest, Dr. Naveen Kumar Patle, Additional Commissioner, Horticulture, Ministry of Agriculture and Farm Welfare, Government of India, who has been playing a very key role promoting plant culture, biotechnology, and also floriculture sector. Also, he is the director of key institution in Northeast. So already all the stakeholders they're aware, and straight away, without taking time, I request Dr. Patle to kindly deliver our chief guest. Thank you so much, sir. What to you, Dr. Patle?
Thank you, Dr. Shukla. Thank you very much. Good morning to all, especially Dr. Purnima Sharma, MD BCIL, and in this occasion, distinguished guest Dr. Rishi Tyagi, Dr. Ravi Kedarpal, Mr. Hon, Dr. Kanchit, and other dignitaries from APARI. So, in collaboration with APARI, BCIL is conducting this program. And I'm just remembering the presence in first webinar conducted by BCIL with the help of APARI. That was on banana, the scope of tissue culture in banana and other fruit plants. Now, today, this is the 4th and final webinar series. And I'm very happy and I'm lucky to attend this program. And I would like to share a few things about floriculture, which is a very emerging industry in agriculture field. The role of tissue culture plant is very much important in most of the crops, especially horticulture crops. But as far as flowers are concerned, the productivity of the plants and especially the quality of the plants, or we can say the quality of the produce depends on the quality of the plants. And tissue culture is one of the very important role when you talk about the quality of the produce. And also from disease resistant point of view, again, very important role in floriculture sector. And there are a lot of advantages when we talk about tissue culture planting material. Now coming to the point, because floriculture industry is a industry in the country, and we are also exporting some products, but again we have the imports in the country from floriculture products. And especially if you talk about the global leaders in the world, the US, Germany, United Kingdom, Netherlands, they are the global leaders. And the export point of view, of course, Netherlands is the global leader in the world. Again, the liking, adoption of the floriculture products in the country is increasing because the business is there, and if business is there, definitely there is a liking of the customers, and there is a lot of choice among the customers who decide the market. If you talk about the flowers like cut flowers, loose flowers, dry flowers, again there are the liking of the customers are increasing day by day, especially in case of cut flowers and dry flowers. So if you talk about the range of products in floriculture industry, definitely now people are coming with the choice like cut flowers, and they need the good planting material if you are going for the commercialization of their floriculture sector. So important is how we can raise, how we can enhance the sector with the help of good planting material and with the help of good management practices. Here the role of scientist, role of researcher, and again the role of our business people is very important, because they are the stakeholders in this sector. As far as the government of India is concerned, we are giving thrust on the sector with the help of our schemes, especially we have one scheme, mission for integrated development of horticulture, and under this scheme we are supporting the farmers and also other entrepreneurs by way of giving various supports like from nursery to tissue culture labs, and also the area expansion of flowers. And we talk about the area expansion of flowers, definitely in case of loose flowers we are supporting for flowers in open condition also, and also in protected environment. Then again, if you talk about the cultivation of cut flowers in protected condition, definitely you have to think for the various types of structures. Protected structures have been designed for various agroclimatic conditions. So the design and also the cost of the structure also affect the floriculture industry, because the cost of cultivation is the important factor if you talk about the viability of the project. So the role of planting material in case of protected cultivation, especially in case of anthurium, gerbera, orchids, and other flowers which are grown under protected structure, are very much important, and we are focusing those commodities especially which are grown under protected environment. So for that we need the good planting material, and we are also supporting the cost of planting material in case of protected structure. Then another point, that this is very important point, you have to focus it on those products which are coming from other countries, because why the products are coming in our countries, because they have the better planting material, they have the better yield, and they have the good quality of produce. So should be also comparable with those products, then only we can also promote our entrepreneurs, our rural youth, and we want to support our farming community in this sector. So, and currently we are also working on one floriculture sector, especially to promote the sector in next 5 years in the way of planting material production, also in the way by way of producing the different commodity of flowers, and also the packaging and drying technology for the flowers. So these are the very important components in floriculture industry. Now, when we talk about the northern region, we have different sectors of flowers being grown in northern region, and again for dry flowers also. In other part of the country, now people are coming for dry flowers, having good packaging, attractive packaging, and they are getting good price in case of dry flowers. But the important is again the technology for drying technology, and again the understanding, capacity building of the farmers, capacity building through demonstrations. These are the very important factors if you talk about the drying technology of flowers. So apart from that, now we have various components. In case of tissue culture labs also, we are giving the subsidy to the farmers for establishment of tissue culture labs. Earlier, the people who established the labs in case of banana and other tissue culture plants. Now the people are coming for the sector also in case of planting material of flowers. So these are the very important thing. Another point is, if you talk about the floriculture industry, then the packaging sector, and also I already told about the technology for to increase the perishability of flowers. So these are the very important factor which have to keep in mind when we talk about the promotion of floriculture industry. So other than this one, the policies, government policies are having very much role if you want to promote the technologies, we want to promote the products. So especially in case of cold chain management, in case of flowers, if you talk about the packaging and transport of the flowers to different destination, again you have to have the proper cold chain management, and that's why we also include the component of our scheme for promotion of cold chain management, cold chain in case of flowers or other products also. So for that we are giving the thrust on our post harvest management of flowers, so that people can take the advantage of this cold chain management, and also they can bring their produce to the destination markets with good price. So these are the examples. Again, in case of flowers, if you talk about the dry flowers, definitely we need to have a good training programs. I would like to request the BCI also, APARI also, because we can conduct more programs on this technology to promote in the northern region and also in other part of the country, so that the startup, nowadays the startup are coming in the industry and they need the proper training facilities, they need the proper capacity building programs, so that they can come up with good products. So in this occasion, I would like to thank the organizers, especially BCIL, who have invited me, and also they have given the opportunity to me to express my views. And at the last, I would like to thank the distinguished guest from APARI and APCOAB for inviting me in this program. And again, I would like to be in touch with you, and we can make more like BCIL is conducting, and in the northern region part of the country and other part of the country, we can also conduct some programs along with my institute, so that the more people can be attracted in this field, especially from planting material point of view, and also from promoting the floriculture industry. Thank you very much.
Thank you so much, Dr. Patle sir, and thank you for taking out time out of a very, very busy schedule. We know that how much busy schedule is there for the Ministry of Agriculture. So thank you once again, and definitely would like to seek your guidance to taking all the initiative further. So Patle sir, we will request for to be with us for some more time, and BCIL has developed a very important tool that is needed by many companies, small companies, because we work closely with them, and what we found that keeping record, traceability, planning the production, it becomes tedious for them. So I request my colleague to just give the brief glimpse of that application. Subsequently, we'll request you to kindly launch this application, this important forum.
Dear industry friends and stakeholders of plant culture sector, we are pleased to inform you that BCIL has developed a unique software application called Plant Tissue Culture Resource Planning, PTCRP. PTCRP is an important software tool for streamlining the workflow. It also addresses a number of pain points of planning and record keeping in day-to-day operation. PTCRP application would include various important modules like media, inoculation, growth room, hardening of tissue culture plant, dispatch, master data management, user management, and most importantly, reporting and documentation. This application would have all the stepwise SOP that has to be followed by any good commercial tissue culture setup, right from selection of plant up to the dispatch and farmer's feedback. The PTCRP application would facilitate systematic automated maintenance of document and record in compliance with the requirement of all the major certification and accreditation agencies including ISO and National Certification System for Tissue Culture Plants NCS-TCP. Thus, this application is expected to address the unmet need of tissue culture industry. Thank you.
Thank you, Mr. Suez. Now I request our chief guest, Honorable Dr. Naveen Kumar Patleji, to formally launch this software application, virtual launching. Thank you so much, sir. So with this now we would move for the technical session. Before that, I would request all the speakers, guests to switch on their camera so that we can have a group photograph before moving to the technical session. I request all the members from Apari.
Hello, this is Jack from Apari. Let's take some sort of group photo. Let's have two. First one, 3, 2, 1. Wait a second, let me save it. Okay, let's take the other one. 3, 2, 1. All right, thank you.
Thank you so much. So now we would come to the technical session. And in this technical session, we have two presentations from research angle, from academia, from research institution, followed by two presentations from the industry. That is success story of ornamental tissue culture. So first, I would invite Dr. Professor Kanchit from Thailand. Is a professor in Maidol University, and he has obtained PhD from University of Hawaii. He has been working for ornamental species for a long time, particularly breeding, conservation, micropropagation. And Thailand is known for the orchid. Even many Indian companies, they are getting their initial material from the Thailand. So it will be very interesting to listen. I now request Professor Kanchit to kindly take over and share your slide.
Yeah. Hi, hello. Thank you very much for introduction. So today I will talk mainly for the orchid tissue culture. Maybe did you see this? Bottle. Oh, okay. So maybe I can share the screen now.
Yes, please.
Okay. Okay. Now, did you
Yes.
see the screen?
Yes, yes.
Okay, my topic is success of ornamental plant tissue culture in Thailand. So first I will tell you the background of the ornamental plant tissue culture development. It starts from the 1967. Some universities start research on orchid tissue culture, and then 1972 is expanded for the business and to over 30 million plants and employ over 300 workers with the 15 labs, and the income is around 2.4 million US dollars. And at present, orchid is still the major crop for using tissue culture in Thailand, but some popularity come up with the new ornamental plants, but those are not stable, depend on the demand in a short time. So orchid still be the main one. So this show roughly, apart from orchid, we have some ornamental plant for export, but the value not so high. This is a value million baht and percent share. These are apart from orchid, which we export around 70 US million dollars per year. So this shows you some variegated leaf of monstera that very popular and doing tissue culture, and also Alocasia silver dragon. And for the journey from research to the commercialization, the research start from the academic institutions, and then development for commercialization by the academic institution and private sectors. So we try to develop low-cost equipment, low-cost tissue culture production, new cultivars, variety or mostly from breeding and know-how trainings, efficient and low-cost cultivation, post-harvest handling, and worldwide international markets. So first I will talk about orchid tissue culture. So we try to develop low cost, and this one show the laminar airflow cabinet made from the wood, which is quite cheap, around 350 US dollars. If we buy from the foreign brand, will cost a lot more than this. So this one can use many labs, even though small labs also use this. And also shaker. We developed 2 stories, 3 stories. The cost is around 850 US dollars, which is a lot cheaper than foreign brands. So also for preparation, many labs, they just use simple. Sometimes you can use from the kitchens, preparing the chemicals and do autoclave. And water, we just use the filter water also enough, doesn't need to use distilled water. And most tissue culture, we start from the young shoot, like this one, dendrobium, from the apical bud and axillary buds. And then we put in the suitable medium, liquid medium, and then get proliferations. And then we get lots of protocolm-like bodies. And after that, we put in the culture rooms and put on the agar mediums, and they form the plantlets. So this one show another small, this one from one commercial unit, Green World Orchid, 4-story, but they can do everything in this unit, like preparation, transfer room, culture room, acclimatization, everything can do in this. So this overall activity in the lab, like transfer rooms. And this one, Mr. Valit Vasiksali, my former master student, is quite successful in doing the orchid tissue culture and ornamental plant tissue cultures. And also Some places they don't use air conditioned room, just evaporation greenhouse. This one orchid can go very well. The temperature is around 28 or 30 degrees Celsius. So this one can save a lot of cost for the electricities and constructions. So this from Bangkok Flower Center Company. So after doing tissue culture, we have to transfer to the greenhouse. So at the beginning, we may do acclimatization, let the plant adjust to the new environment, which have higher light intensity and lower humidity for a while, maybe 2 or 3 months. And then we put in a pot. This one, they are preparing to go Phalaenopsis in the northern part of Thailand. Later on at the blooming stage, they will export to Japan. So the cost for production is a lot cheaper to produce in Thailand and then export during the blooming time. So we use sphagnum moss. This also sphagnum moss, and put the plantlets in, and put some foam block for aeration and drainage. So this one, the plants. So some we use a lot of harsh coconut fiber for the plantlets. Orchid breeding is also indispensable. Orchid, 2 things, tissue culture and breeding. So we cannot live without these 2 things. So new cultivar, outstanding one, should come out very, very fast to support the demand. So we start from pollination. And this one show one Catria hybrid. Amaki, this one really good for corsage. You can see the ancestors are from different color, shape of Catria groups. And then when we get the outstanding selection, we do the tissue culture to produce a lot of plants, to sell the plant and flowers. So also some dendrobium cut flowers, many shape and colors, shapes, and different colors. Vanda also, we developed the new cultivar that can grow very fast. Within 2 or 3 years can start flowering. Before need around 6-7 years, and also year-round flowering also. So we get lots of production and high production from Vanda groups. So doing tissue culture is not only one part. We need to do the production technology to support the tissue culture. Once we produce the plantlet, we need to grow them well with the low cost of production. So in Thailand, we developed the orchid production technology, like the greenhouses. So mostly we use the paddy field because we get full sunlight and the land is cheaper than in the city or nearby city. And also nothing block the environment. So people mostly go to the rice field and make some dam to prevent the flood. So at the beginning, 40 years ago, we used a bamboo for the shading and bench, which are not long lasting. So we developed to use the saran or netted cloth. Like today, if you go to Thailand, you can see all these black netted greenhouses. So these are some vandas growing in the saran house. And mostly now we use a plastic basket, which is quite cheap and lightweight. And if they have enough relative humidity, we don't really need some planting materials. So these are dendrobium cut flower. We use coconut husk. And sometimes we put in the coconut husk in the plastic and have the tray. And watering also, the best way is to use the manpower to do this, like use nozzle. But if we have the larger size, we may need a sprinkler. And this one to spray pesticide and fertilizers. For planting material and containers, in Thailand, mostly we use the coconut fruits because it's cheap, available everywhere, and give good growth of the orchids. So we make different shapes for different sizes of orchids. The last one is the block for cut flowers. So this one, coconut hush for the young dendrobium. And some farmer, they use a foam instead of throwing away the foam box. You can use to support the plants, but we need more water and fertilizers. So the foam block doesn't block the roots. The root can penetrate to the foam, and so the plant can grow quite well and lightweight, and we don't need to waste many things in the environment. And for cut flower, we don't need a good container, so we just go on the coconut block. This one allow 1 foot times 1 foot. We put 4 plants, and when the plant becoming larger, we just take out alternately. And some farm, they use a cement blocks. This one will last forever, but you may need to water more and more fertilizers. This one, many farms start using this instead of coconut block. For pest control, we develop many recommendations for control by biological control or chemicals, and for post-harvest and packaging technology. So after we cut the flower from the farm, we need to maintain high humidity and give some food. And this one, they are sorting out and preparing to send to the local, maybe to some provinces, like the plants and flowers. And for export, we already make like corsage or some arrange already. When you reach the destination, they can start using all these orchid products. And for orchid trade, in Thailand, many like local farm, they have like supermarkets. People can go and buy plants and flowers. And for the growth of the business, Thailand, we export orchid around $70 million from the year 2007 to 2019. And this one showed, the left one on the top is a orchid in the flask for export from tissue culture. The right top one is sending the plants, and the below one is for cut flowers. These are for export. For orchid training very important because we can disseminate the knowledge to the farmer, private sectors, and so they can expand the orchid industry. This one in the room, lecture room, many professions come to learn orchid production and tissue culture. And in the lab, we do seed germination, tissue culture. And also in the greenhouse, they do the transferring the plant from the bottle to the pots. And also they have study tour to visit the successful farmers. You tell how he do all the business and technique to grow orchids. So people can learn a lot. And orchid show also is important. So you can see the market right now, how many cultivar they like, and people can meet one another and can exchange the plants and have the customers at the orchid shows. We have orchid show for many associations, many times a year. Like this one is a big one. This is an entrance to the orchid show, and they have some landscape to show the new cultivars. So for the secret of the success in Thailand, because we have favorable climate and availability of good quality water in plenty, leadership in adoption and popularization of orchid cultivation, richness of indigenous orchid genetic resources for crop improvement, and also improved tissue culture, production technology, greenhouses, containers, post-harvest processing, quality control, packaging and transport, and their application in orchid trade, and efficient communication networks and international acceptance with maintenance of standard. So acknowledgement, I would like to thank the APARI, BCIL, and COCOP for invitation, and Assistant Professor Dr. Supat Kritirat from Faculty of Science and Technology, Nakhon Sri Thammarat Rajapat University, and Mr. Walid Wasiksili the owner of Green World Orchid, for providing the information and some slides. Thank you very much for your attention. Thank you.
Thank you very much for Professor Kanchit.
Yeah.
I would say that is kind of visual treat for all the audience. Excellent work and such a nice way of presenting your work. All colorful photograph, flowers and orchids. Thank you so much.
Thank you.
So before moving ahead, we have a just like a previous webinar, we'll conduct a brief survey. Just will take few minutes. I request my colleague to project the poll questions on the screen. I request all the audience to kindly participate. You are getting 3 questions, and for all the questions you have multiple choice. Just select one. Like the first question: have the plant tissue culture technique been optimally deployed for the production of ornamental plants in your country or region? Please select yes, no, or not sure. These all feedbacks are very important for us as organizers. Now, the second question is: do you agree that there is a need for an effective training program for promoting entrepreneurship in tissue culture of ornamental plant in Asia Pacific region? Option yes or no. And the third question: do you agree that a user-friendly software, just like an ERP, would be helpful for operation, quality management, and traceability during the production process of tissue culture raised plant? So these are 3 questions. We'll just take few seconds to complete this thing. So we can wait just 10 seconds more. Then we'll close this poll and we'll show the result to the audience. Five, 4, 3, 2, 1. I request my colleague to project the poll result. Okay. So 67% they are saying that tissue culture has been optimally utilized. So there's big gap. Still, there's a lot of work need to be done for making this application in the area of plant culture. And 100% they're saying there is a need for training program. Very important for BCL and Apari. And Dr. Tyagi, I think we should formulate some kind of program with the help of Apari and the regional partner for the training on the ornamental tissue culture. Then the third was 82%. They're saying that ERP or PTCRP like application would greatly help them in maintaining the record, documentation, and traceability. So thank you so much to all the audience for participating in this poll. Please stop this result. So now we'll move to the next presenter, Dr. C.R. Aswath. Dr. Chenna Reddy Aswath, he's a principal scientist and head of the division of floriculture of one of the very important institute in India under ICAR, Indian Institute of Horticultural Sciences, Horticulture Research. Dr. Aswath has done PhD from Guwahati University, and he is recipient of many awards like Dr. B.P. Paul Best Scientist Award, Sub Award of Excellence in Horticulture Biotechnology, and he has worked in a number of crop species and mainly on the flowering plant and ornamental. He is also a recipient of very important fellowship, Brainpool Postdoc Fellowship from South Korea, and is a member of various prestigious committees. So we are very pleased to say that we have an eminent expert with us, Dr. Aswath. Over to you, sir. Please share your slides.
Thank you, sir. Okay. Esteemed panel members and the participants, you can see my slides.
Yes.
Okay, thank you. Yeah, right. Coming to the floriculture, because I'm a floriculturist by profession as well as even as a farmer. See, floriculture is completely, like production of loose flowers, cut foliage, potted plants, even dried flowers. Now India is getting 50% from dried flowers. But for all this, now I found that it is the seeds and planting material and the tissue culture plants which are most important. If you fail in this plant tissue culture plants, if you are not producing it, we cannot produce proper cut flowers. So especially in the foliage plants also, very, very important, good mother plant that is coming from tissue culture plant. So I think this is one of the important area where BCL has taken up, and probably this is the base for our entire our production system of the floriculture. Yeah, coming to the global plant tissue culture, it is almost $382 million dollars, and definitely India is going to take up in a big way, because I have seen for last 30 years, India is leading in Asia Pacific. Of course, now it is around 500 crores. But I have seen, like labs which are just even 0.1 million plants to up to 20 million plants also is there. So it is it's a vast country. It's a huge country where thousands of labs are there with a very, very small, like 1 lakh plants, but they are really contributing much to the production of the planting material, because they are the owners. And it is right now, it is required to encourage this industry, whether it is a small or big or medium, whether it is 1 million or whether it is 20 million or 500 million, it is required, like in a big way, we have to encourage them. So definitely the Bangalore is one of the darling air cargo, which almost sends 41% to these countries. Yeah, in fact, I read one of the meeting proceedings where the exporters, they had a meeting, EPIDA and the DBT, they had a meeting of exporters, and they have told these are the problems. Yes, I feel because I worked in industry also and I worked in academia, the power tariff is one of the biggest thing which, like the tissue culture industry which is paying. And some state governments, they are putting the commercial tariff. And this is where really we have to discuss in a policy policy matters. It is a policy matter. The BCA has to take up the government of India. The tariff is the one which really kills the industry. Then definitely the skilled workers. After work for 2 years in South Korea, I found really our workforce is very, very underskilled, and this is where ICR and universities has to take up to increase the human resource. And of course, contamination problems, they are the local. And the transportation. See, this is another big problem, because we also now we want to send gerbera plants from Bangalore to Northeast. We are really finding difficult, because they are very expensive if it goes by flight, and they don't sustain when it goes by a train. And a lot of research is required, like in the packing and even other things also. Then objections raised by forest and quarantine departments. Again, it is a policy decision. Yes, many of the labs, they come to IHR for a quarantine certificate and many certificates. Recently, when aquatic plants lab came, and they're telling that they need a certificate that these plants are not wild, because they are the certificates which are asking by the forest and quarantine. Probably these two things, the tariff and the aquatic plants, has to be discussed in the policy decision. And the exporters felt that we have to now showcase. It is the time we have to make a sort of exhibition, and we have to showcase our plants. And this is where I request BCL that definitely we will be there. IHR will be there, and if you go ahead with a sort of exhibition or a sort of meeting with the exporters. Then I was just listing out, like industries. There are so many industries in around Bangalore and down south. Definitely, there's a lot of industries, like internationally recognized tissue culture industries like Cave Biotech and Floratech is there. But there are many small scale industries, like it is just only with a 2 or 3 laminar airflows. So they are also to be identified, and that is where I feel that the small one we have to really encourage, because mass production is one thing, and production by masses is also very important thing which we have to think about it. Yeah, coming to the actual topic, like commercially applicable methods. Yes, we are now most of our labs and most of the research has been done in organogenesis, and definitely a lot of plants has been already standardized. Very good protocols are available, but right now we have to shift it to the somatic embryogenesis, because this is the one which really in future it is the one where we can really go for mechanization. And of late, even when I was in South Korea, I met one scientist during net. He has worked extensively on thin cell tissue culture. This is one of the technology which is not really picked up by the labs, Indian labs, and this is where again I feel that like we require more of more training. Then off late, bioreactors are coming in a big way. See, during 2000 only, I saw that a huge bioreactors in South Korea producing ginseng and lilium bulbs and other things. So in India, still we are in infancy. And of course, we tried some orchids and anthuriums and liliums, and we found it is very encouraging. And this is where, like academia now, we have to work and encourage the industry to go for it. Yeah, slowly I'll just go about it. Organogenesis. See, organogenesis, it has got a lot of advantages. It is a low concentration of plant growth regulators, and it minimize the risk of somoclonal variation. But see the disadvantages. See, suppose a startup company has to start a small startup company in tissue culture. It takes a lot of time in the initial time for the induction, for like establishment. To come to the subculture level, probably it requires around 8 to 10 months. So this is where I request the industry, because a lot of industries are there, BCL is there. Now we have to have a sort of labs where they have to sell the initial cultures. So that's right. I can take, say, I can take a caladium cultures, I can take a Dracaena, I can IV cultures from a lab, and I can straight start my lab. So now if a person who starts a lab, it takes at least 1 to 2 years to establish it. And this is where, like the big industries really has to support the smaller one. And we have to have a sort of a culture bank supported by the government, or someone has to like bell the cat. And this is really my humble request. And of course it is cheap and quick also. So a lot of these cultures are now exported from India, like gerbera, anthurium, orchids, hosta. They're all like established cultures, established industries. They are doing it. Because tomorrow if I want to send an hosta, it is very difficult for me to initiate and send it. And this is where like many of the industries, they are doing well. Whether it is chlorophytum or ficus, there's a lot of demand is there. I in somewhere in one report I heard that it's around 3 trillion plants demand is there from the Europe countries. So India is a vast country where we have a like from small to medium to big industries are there. Definitely we can take a lead in exporting these plants. Yeah. So as I said, cost of transportation. This is where, in fact, many of the times we failed to send our plants from Bangalore to Bangalore to Northeast, because its flight charges are very expensive, and lot of things has to be, we had to maintain the cool chain and all. So this is where, in fact, Jain industry long back they have come out with, preparing the plants at the end of the plants in a sort of sterile cover, so that the entire cover can be sent it through a train or through a transportation. But I do not know, like afterwards, even the academy also has stopped. But this is where again, we have to really think about it, sending the plants in this type of pouches across India, so that it's easy to transport also. And I normally visit many of the industries, especially in subculture time. I see, like the plants, normally they just cut it. They just cut like this. And we found that, like a lot of vigor will go off when they really cut it, and a lot of callus will form, and the plants are, like amenable for infection. So we found that there is one, like small method. It's called nicking method. You just nick a plant, a clump will come out, and that will give you the best plant. And this is where, in fact, we are training many of the, like trainees who come to our labs. Then coming to the somatic embryogenesis, I really feel that the somatic embryogenesis is the one which really future matters, because they are the one where large scale production can be, like we can do it, and even mechanization can be done it, and even we can produce the virus-free plant. And you can see here, even when they say, suppose an industry, a person is producing, say, suppose some orchids. If the demand is not there, somatic embryos can even, we can send them to a sort of dormancy, and we can take it out after 6 months or after 1 year when the demand comes. So this is where we are also working out, and a lot of research is there. And we have to slowly now we have to shift it from orgogenesis to somatic embryogenesis. Of course, some disadvantages is there compared to the organogenesis, but still a lot of advantages is there, so that the industry can slowly shift it. See, there are a lot of good papers are there where even the rows, you can see somatic embryos, they just came to the field and it is flowering very well. So it is like a natural, I mean, like a synthetic seed, which can go in a big way, and easy to transport, again, easy to send it across also. You can see here thousands of plants can be sent you just through one small pouch like this. So there is a lot of, scope is there to go for a somatic embryogenesis rather than from the organogenesis. So even we can use it for, some sort of mechanical transplanting also, we can use it. Then bioreactors, as I exposed 2 years in South Korea, it is really, it is coming in a big way. The proliferation rate is very high. Labor is very cheap, because one person can handle entire lab, 1 or 2 persons. And a lot of, mechanization is there here, so that we need not bother much about the pH, EC of the media. Everything is, completely, it is mechanized. So of course, 1 or 2 disadvantages where if entire reactor is contaminated, it will go off. But there are a lot of success stories. Probably NRC Banana now has come out with a banana bioreactor where thousands of plants can be grown, just in a flask. And we have worked with Lilium, and we got really good advantages. So I have seen from 5-liter bioreactor to up to even to 20,000 liter reactor. And we have standardized most of the things in our 5 liters bioreactor, but we want to really, require funds to go to upkeep or to go for a bigger bioreactors, which is really very, very infant in India, which is again a need of the hour also. It has got a lot of, advantages. We can really maintain completely temperature, pH, concentration of oxygen, everything. And you can see the economics. Of course, this is in ginseng, 2 tons, it takes around 1 year. Whereas in 2 tons, it requires around 7 years for 600 hectares. So there is a lot of advantage. Probably we may have to shift, technology innovation, where India has to lead by technology. And this is one place where really we can think about it. Then this is, of course, our we worked on Anthurium, and it is really working very well. You can see thousands of protocol like bodies even in Anthurium we could able to produce, which normally Anthuriums now they are produced through organogenesis. You can see beautiful PLBs, just like arcades even in Anthuriums also. And we worked, of course, in Phalaenopsis also. It is working very well. In fact, I bought one small orchid, bioreactor from Korea, and we tried so many crops, and it is working very well. So and Lilium also. See, Lilium almost takes 1 year, like a production of a bulb. And this is where, like in the stage 2, we wanted to develop through the bioreactor. And we got really success. One of my PhD student, he has produced Lilium bulbs, and we could able to produce Lilium bulbs, and we could able to see when it is going to produce, how it is going to produce in bioreactor. And in 3%, 6% and 9% sucrose, we found, like the Lilium bulbs up to 20 grams. So now one of our student is now working out from increasing from 20 grams to 40 grams. Yes, again, this is one of the leading technology, which definitely it will pick up in after some time. Then chrysanthemum, we have standardized it. Thousands of planting material can be, even with a sugar-free medium, we can just produce it. So, of course, jewel orchids in South Korea, it has already been standardized easily. And gerbera. See, gerbera is the one where I'm really interested, because we have released 4 or 5 varieties where gerbera normally, like 40 rupees their plant is selling, because of their tedious, subculture, frequent subcultures and all. But when we really go for it, you can see here, for 1 hectare, it costs almost 19 lakh rupees for a farmer to go for gerbera plants, because he requires around 60,000 plants. If you really bring down the plant material, we can just, with 7 lakh rupees. So of course, we are working out in gerbera, and we got some success in gerbera also in bioreactor technology. Then, of course, we found that a lot of, problems are there, whether it is a low cell productivity or genetic instability. But what I suggest is industry in India, industry is the one which is not really supporting the academia. I have seen in South Korea where completely my research was supported by, sort of industry there. Korea Vision Biotech has supported me for my PDF, and I could able to do some transformation in long grasses. But in India, that is really lacking, and that is where, we have to go in a PPP mode. Not encouraging, really, not encouraging. For last 30 years, we are talking it. So far, I'm going to retire. Not really, like industry support is there, other than the biotechnology, DBT or some ICR. So then TCL technology, as I said, it is highly homogeneous tissue which we are taking it, and they are really, like they can make a sort of thousands of plants can be produced in a single homogeneous tissue. And even if we can eliminate the virus also, large number of planting material is possible. You can see here, there is one Duyang Tannet. He has come out with a thin cell layer culture system, a very good book where all protocols are there. In fact, I'm also one of the authors for 2 chapters in this book, which is by Springer, and he has come out with, like complete protocols for most of the crops. And you can see here the number of plants, thousands of plants, just in one small TCL layer, which is about 0.1 mm. So either transfers or, like very good protocols are there. Again, like it is still in sort of labs only. Again, industry has to adopt this type of system after getting trained from the academia. Okay, you can see here, like a number of plants which, in Saint Polia and other plants, the African violets. It is very, very, like, of course, it is highly technical, and it is in Lilium, thousands of bulbs can be also be produced using a TCL technique. So when it comes to the, like some sort of hardening, I found that, like most of the Indian labs, like we are really not doing very well when it comes to the exagar plants, like hardening of the plants, because we never bother about how much humidity we give, temperature we give, then media, then what type of water we give. And I have seen, like there is a concept called reclinable polyhouses where we have to expose the plant even to the higher temperature, lower temperature, depends. Because normally I have seen the polyhouses where they just put a shade net of 50%, and, like a completely 50% shade net always will be there. See, now in Bangalore, last almost for 1 month, there is a cloudy days. And during these cloudy days, the plants will not really develop very well. They will not open up their stomata. So this is what is called a reclinable, where whenever the light is not there, they should automatically open so that the more light will come, so that the plant will get more proper hardened. Because I have seen, like whether it is a big industry, I don't want to name, they give the plants, and when it comes to the farmer, within 4 or 5 days, the farmer will lose 20%, 30% of the plants. It is because of the problem in hardening, because they are hardened in a very, very sophisticated environment, which is not really not required. So it is the light which matters a lot. It is the humidity which matters a lot. It is the temperature. So these things matters a lot, and this we have to take care whenever we harden the plants. Of course, we have worked a lot, and now, in fact, we have come out with a sort of vertical structures for hardening the plants, where you can harden the 6,000 plants instead of 1,000 plants in the same structure. So yeah, I mean, in a polyhouse, if you are hardening 1 lakh plants, in the same polyhouse, we can harden almost 6 lakh plants using this type of structures, where uniform light will be there, uniform humidity will be there. Everything is uniformly we have maintained it. So this is one of the, like a sort of wonder for a small persons. With just in a 60 feet by 30 feet polyhouse, he can almost or 1 lakh plants, he can harden it. So this is the standardization which we have already done it. And if anybody is, like we can definitely train them. Then in tissue culture labs, what I found is these are the 5-6 problems which normally people, like they face. But most of the time they will not discuss. They will throw the culture. They say some problem is there, so they could not. So the first thing is, of course, the recalcitrance. Say recalcitrance is many times, like the plants will not grow, but still they subculture and they root it and they give it to the farmers. I feel, like really many of the anthurium plants we have seen, they're highly recalcitrant. But they just root and give it, but they will never grow. They'll never grow when it comes to the, like a farmer's field or a grower's field. So there is a lot of, like science behind, like recalcitrance. There are a lot of free radicals are there. Of course, now there are a lot of, we can eliminate the free radicals in the media itself. And this is one thing, like people say that they change the media. They change the agar, they change the growth hormones, telling that of course they are not growing it. It is not that, like a lot of science behind it. There is a, there are a lot of papers also. There is a juvenile tissue we have to take, very vigorous tissue we have to take. And if juvenile tissue is not there in the initial explant, we have to initiate the juvenile tissue and go for it. Then of course the contamination is another big part. Of course, the many of the industries, they have closed because of the contamination part of it. It is really, like a local. And we have worked a lot on endophytic contamination. In fact, one of our scientists, one Dr. Pius Thomas, he has got more than 100 papers in international journals where we have worked a lot on endophytic contamination. In fact, endophytic contamination, endophytic bacteria are very good. They give a lot of, symbiosis to the plants. But if the proper, temperature is not there, if a proper facilities are not there to plant grow, this endophytic will just come out of the plant. So, and it, it destroys the industry. So there is a lot. Of course, now lot of industries, they use ppm at the end and they just give it the plants. But initially, if you use ppm cultures, definitely the cultures will not grow properly, because it, after all, it's an antibiotic. Some people use cmit, which is again dangerous. So there is a lot behind, again, contamination also to study. There is a lot of science behind it also. Then phenolic browning, especially the people who are working on callus, again, it is a problem. So we have worked a lot. Many of our students did PhD on this phenolic browning. Antioxidants are the main, we have to, now we are using a lot of antioxidants like PAL and even PVPs. And a lot of papers are there, a lot of good papers are there. So definitely the industry can utilize these knowledge from the scientists. Then vitrification. Vitrification is again very big problem in especially herbaceous crops like gerbera, where, they put a lot of BAP, where fragile plants you can see here. These plants are the one which, when it comes to the farmer's field, goer's field, they die because they're all fragile. Their stomatas are not well developed. They're completely open. So they are the one, a lot of things is there, because we don't follow a proper vitrification procedures in our labs. See, these are, even the matter, it matters what is the type of container you use. Albinisms will come, dwarfing is there, vitrification is there, depending on the growth of the containers. So a lot of studies are there. Even I never seen any industry in India, they use, cooling of the bottom. Whereas I have seen in South Korea, cooling of the bottom is very important so that the humidity will not stay and the contamination will not come. So this type of, even hyperhydricity is because of the, we are not cooling up the bottom. So there are a simple small procedures where, if you follow it, this type of problems will not come in our plants also. Yeah, of course, like these bottles, you can see here, completely there is down, there is a cooling is there. So that is why, they don't get a sort of vitrification compared to the one which earlier I have shown, as a solid like this. So, so what I found is, now we had to follow some protocols where short and medium conservation is, we have to develop, because there are a lot of demand. Sometimes demand will be there, sometimes demand will not be there from the consumers. So when demand is not there, we have to properly, conserve the mother cultures. So that is a problem, mother cultures are not properly conserved. That is why, endophytic bacteria will come out when they're just reviving it. So this is one of the important, thing which we have to follow it in the labs, to how to conserve whenever the demand is not there. Then mutation is another thing which, most of our Indian labs are not utilizing it. See, a lot of, planting material can be induced, mutated, and we can really sell it as a new varieties in Indian labs, which are not there. And now I found that even lot of planting material, especially the cuttings, is a big industry in India now. See, now the carnation, here, I heard around 5 million plants is exporting the carnation. It is a simple procedure, taking the cuttings, just put it in plug. Of course, it is like a tissue culture where instead of MS media, they use a media which is amenable for these cuttings. And this is also requires a lot of, support from the government, training in the terms of training, which is really required, to go for a startup company. So I have few suggestions for the industry, as for the academia, and of course, for the BCIL and the panel members, I mean, the policy members. See, industry, like we had to now, industry has to help the academia, especially. See, we cannot keep on copying it there from the foreign. So we have to now, biosynthetic roots, we have to see. So a lot of science behind, like plant, when it plant come out. So we have to study. So industry really, help the academia. Then we have a lot of Indian native ornamentals, which we never really tried. We all think about only carnation, gerbera, and these only. But there are a lot of Indian native ornamentals where we can really exploit it, because so far, like we are not really exploited. This is one thing which now we have to think about the native ornamentals. And now big companies, there are big companies where really they have to support the smaller companies by outsourcing the production, because they see, always I have seen, like Ramco Biotech, they could not sustain because a lot of overheads will be there. So unlike, you take any industry, a car cannot be manufactured in only in one company. At least there are 15, 20 companies which supports the one car company to come out. So like this, here also we have to have a sort of big industry as to, they have to explore of outsourcing it to a smaller, so that many people can, it can be, mass produced, I mean, produced by a mass, so that an industry, a final industry, one can just take it and export also. So one industry instead of one industry, I always feel that, we have to involve the Sri Shakti Kendras. There are a lot of good rural youths are there. Now, in fact, ICR has come out with the RA program where a lot of youths are there, where we have to train them. In fact, now I proposed a project in DBT, which I could not get it. Why don't we have a sort of hardening units in different districts where an hexagon plant can be sent there, where he can harden it. Instead of all this, say, suppose now if I want to buy 20,000 plants from Pune, KF Biotech, all the way I had to bring it from Pune. Why don't we have a sort of hardening unit where I can buy an hexagon plant and harden in the itself? So it is now, it is the time where we have to require a sort of industries has to come with a small, like hardening units. The big industries has to support, give them the hexagon plant, so that they can start hardening it and they can distribute among the farmers or the growers. Then, of course, so develop the tools and techniques that are most convenient, efficient. It is required. Now, in fact, tissue culture can be done even without autoclaving also. There are Japanese technologies which has come out. Even the tissue culture can be done without autoclaving. So it will become a small industry in future, definitely. When the days to come, a small woman can definitely, produce thousands of plants in her, like a small bedroom, and then send it across. But it is all really now, we have to now get up. We have to have a sort of human resource development in a sort of training. We are doing it, and it should come in a big way, because batches and batches of skilled workers we have to now produce, whether it is a budders, whether it is a grafters, or whether it is a cutters, whether it is a, like a greenhouse managers, all this as human resource development. And that is where our Prime Minister is stressing upon. And I really thank BCL to give me this opportunity. I am very, very thankful to Shuklaji for giving me this opportunity to speak few words. Thank you very much.
Thank you very much for Dr. Kanchit for this very informative presentation. You have provided various insight, not only for the technology, for the commercial aspect. Also, one important aspect you highlighted, support to academia by industry. I think that has to come up. And just wanted to inform that BCIL has been identified as a technology transfer office by the Department of Biotechnology. And that is one of the mandate to support academia through industry. So definitely would like to explore this possibility under your guidance. And also you highlighted that local species that has to be promoted. And many small companies, they can join ahead and they can create a good ecosystem. So we noted all your suggestions. Thank you so much for this nice presentation. Thank you once again on behalf of all organizers. Thank you so much.
Thank you.
So we have second round of poll. Just we have two questions. Let us please answer the slide. We already flagged the poll 2. There is just two questions. You agree that tissue culture of ornamental plants has the potential to boost the economic growth of the country at national and international level. So you have to select one option out of given 3 options. And the second one, are quality management practices beneficial for improvement in the production process of superior tissue culture plant of ornamental? Please complete your poll in few seconds. We'll just wait for 5 seconds, then we'll close this poll and announce the result. 5, 4, 3, 2, 1. Please close and press the result. Okay, 99% they agreed that tissue culture of ornamental has good potential to boost the economic growth. And the second one also 99%, they're saying that quality management practices are essential to produce the quality plants. And the quality management practices, already our expert, they have talked about virus indexing and reducing the various kind of technical constraints, including hyperhydration, contamination, mortality. So with these quality practices, by adopting good SOP, I think one can address those challenges. So thank you so much. Please unshare this poll result, and we'll move to the next presenter. Now we have completed the academia presentation, and now very interesting session will initiate now. That is the session by the industry, and we had 2 eminent industry experts in this session, and very successful company, and they will talk about their success story. So first presenter is Dr. Mr. Onyan Zhao from Taiwan. He's director and the president of Grand Biotech Company Limited, located in Hsinchu Science Park, Taiwan. And this company is doing excellent work. In last 30 years, they have exported plant to Japan, Netherlands, along with the providing planting material to the country in domestic. Mr. Zhao has started this journey as a scientist from Taiwan Seed Improvement and Propagation Station. Then in 1989, more than 30 years before, he started this GBC, Grand Biotech Company. And he has managed it very successfully. And his experience will be very, very beneficial for all our participants. So Mr. Zhao, over to you.
Okay, thank you for your introduction. We are very impressed for the tissue culture plant production from Thailand and also from India. I think in my lecture, we will focus on the supply chain from Taiwan to the Netherlands and other part of the world. Well, can you see my presentation now, everyone?
Yes, yes, very much.
Yes, good afternoon, everyone. Ladies and gentlemen, I'm happy to meet you online. I'm Zhao from Grand Biotechnology Company, Taiwan. We located in Hsinchu Science Park. It is said the Silicon Village of Taiwan. The mega company said Taiwan Semiconductor Company is one of my neighbors. The most orchid is our theme of today. Become the biggest turnover rate of the pot plant since 2003 in the US. It become a worldwide pot plant. In the next 15 or 18 minutes, we will see how tissue culture companies achieve an international supply chain growth by world war for the last 30 years. I think we have some mistake.
Any
Sorry.
difficulty you are facing in moving the slide?
Yeah, I think something wrong. I share again.
Yeah, that will help.
But I cannot see mine.
Yes,
Is that right?
first slide is there.
Well, I cannot see how to move it. Yeah, sorry. Okay. Today we were discussing 3 topics for the spec and the supply chain issues for the OKI supply chain. First, the OKI supply chain, why Taiwan's matter. And the second, the spec of most OKI supply chain. And the third, the spec of plant-tissue fab. I think the fab is a word of the factory and the lab. The first topic is the most supply chain, why Taiwan's matter. The most OKI supply chain, we have 3 sections, why Taiwan's matter, and who's who in Taiwan company. And the breeding most OKI supply chain. Why Taiwan's matter? This is a Business Week report at May 2005. The story says the global economy could not function without Taiwan. But can Taiwan really peace with China? They discussing the supply chain for the electron products between Taiwan, China, and the world. The current situation of the US and China conflict is the same story as 18 years ago. Who's who in Taiwan company? Here's a gallery of the island top company leaders and where their company located in the supply chain. Taiwan is a tiny island. The size is almost the same as the Netherlands. Taiwan will play an outsized role in the global economy. At the time, 2005, there are 2 highways connect all companies in Taiwan from north to south. And now, the one-day life cycle achieved by the high-speed rail, the same as from north to south. The supply chain of semiconductor company industry is going to be exciting. In Taiwan, we have many farmers. They work for most of the in Taiwan. They are not only farmers, but crazy for any unique moss orchids. They love all different types of moss orchids. They are dedicated in moss orchid breeding for what they love. So, someone like Joint Orchid Nursery dedicated in Big White, and Fuller Orchid Nursery dedicated in Hurricane type, and multiple types from famous Brother Orchid International. Those farmers not only share the pollen, but they also share hybrid flowers to each other. That's why anything is possible to get so many innovation hybrids last 30 years. Every March, Taiwan International Orchid Show you the latest closing performance in Taiwan. Due to the COVID-19, the International Orchid Show was stopped for last 2 years. We can see the big white and the big purple, and also the multiple and the medium type of orchids. All we select and make it commercial propagation, make it commercial production in Taiwan. Say this is a commercial production at an OptiFlow in the Netherlands. Our supply is non-stop for last 20 years. It is says a commercial scale production. The most orchid supply chain is complicated and long term. All work start from breeders' innovation to make a clothing. They breeding for the hurricane and the breeder for big white and also yellow and Bigfoot, for example. Breeders send their hybrid seeding to grow at 1.7-inch pot. For grow A, for grow B, grow C, or themselves. After 26 weeks, the finished grower are growing to after defrost, and they were sent to the breeder. All spiking, most of the excited growing plants go back to the breeder and the selection. The best commercial potential plant move to tissue culture lab again. Clone for next testing. Go to breeder have all our breeders have a very secure connection with tissue culture lab always. After selection, the plant will go to testing evaluation for next 1.5 years. If we can find good plant, they will go back to the tissue culture fab again. It is a commercial scale production. The best commercial potential plant will send back to the tissue culture fab again. It is a time for breeders or the young plant company in the Netherlands. They have started to the latest promotion their latest production. We can see tissue culture fab is an essential player in the most office. We show you the time frame for commercial most office. Mega crossing, it is essential. It is wait 6 months. Mega crossing, it costs about 6 months to get a seed pod mature, and hybrid seeding grow to the first evaluation. It costs another 3 years. Crown plant that grow to second evaluation, it comes to another next 3.5 years. At the start of commercial production, it is cost about 9 years was gone. The time frame is very important to see. It is very complicated and very long term to see. Our second topic, we're talking about the spec of most office supply chain. We have 3 sections too. The spec and the spec for most office supply chain. And we review the iPhone supply chain for everyone. Or you have a smartphone, you can connect the world. That is a smart. What is the spec? Help this important work for you. As we know, GSM is popular spec for most of the country. When you travel in Japan, you have to change your smart card, your SIM card to connect with other smartphone in Japan. It is safe, the Docomo spec for Japan only. So you know the spec is powerful for the world. It is life and death for a company if we go wrong the spec for our products. There is a CD and a DVD disc disappear last few years after USB disc, for example. We come back to our most office supply chain issue. Yeah, the photo is Mr. Wu, the founder of Oxfam in Taiwan. He told me the most office is a cash product 20 years ago. He transferred his tissue culture plant to 1.7-inch pot. He smoked it, and after 2.5 and then 3.0 inch, and even for 4.0 inch. It depends on when he expects to sell his plant for sale. His most office could flow at any pot size if necessary. The most important, all stage of this product can be sold for cash. The plastic pot becomes the spec of most orchid trade in Taiwan. Farmers buy the plant is called by the pot size and also the plant age. All pot size have a couple tray for easy operation purpose. The Prague system, it is a new system successfully introduced to most offices around 2014 and 2016 in the Netherlands. My customers in the Netherlands, they are always happy for my quality of plan system. There are almost no loss from frost to greenhouse. And the most young plant growing speed is faster than compared to other systems. The growing spec is changing, and the most office price is changing rapidly. This is a supply chain chart for the iPhone. We know the iPhone is start from the US, California. There are total 31 countries and over 750 companies involved in the work of iPhone. The last assembly work in Shenzhen, China, and after shipped to the world. Now the assembling work also work in India. Every company is essential for this supply chain. If you are in, you cannot go any mistake within a supply chain. That's a powerful of a supply chain make the business bigger and bigger. Well, when we Google the US-China trade war, for example, the Google will show you so many different scenes with the trade war. We can see in the central, in the core of this subject. The container is the core of subject. Does it mean the international supply chain is dominated by the spec of the container? The last topic is the spec of tissue culture fab. We have two sections for this topic. The spec of carrier and the trade, and the environment control spec. I think this is the standard operation procedure for most of the tissue culture in the school. It is always very easy work for small volume in the school lab or research lab. For the commercial volume production, I think it is a state of art. Quality, reliability is essential for all fabricants. This is the nursery for my custom Pelat orchid in the Netherlands. They have millions of plant production start from many tissue culture factories. All tissue culture factories have to ship quality and plant on schedule. Different boxes means come from different tissue culture factory. Every box have to input many knowledge for the plant growing region. My company starts from the screw cap. Container 30 years ago. It is perfect for almost all plants we can grow well in my factory. The only bad is hard to open and the screw cap and the recycle. The band container is come from Ushepa companies in Netherlands. It is perfect container for most of the production, but the supply chain from the Netherlands consume too much carbon footprint to me. We have the PP type container tall and the short one in our spec. All middle size, special for the most what design. We name is T4. Let me show you the plant growing performance change between the container. You can see every week the plant growing is different. The young plant performance T4 spec is much better. We can see that it is much better compared to the PP spec within 12 weeks. So our customers, they could decide the week number for shipping, which is the right time for their nursery. I think all is possible under our control technologies. My culture room is always under control. Its temperature, lighting period, and humidity. The spec of plastic tray supports the boxes and the cultural shelf. It is not only the lighting and it is easy to handle, but it is a unit for the calculation of our production. We are ready to introduce the automatic workflow to the next step. Our operators work hard to growing planet and to be unique, to be our plan to be uniform. Everything comes to end and finish production nursery. Reliability means our schedule and the quality control. And I think we are the best propagation partner for all of you. You can book your setting and the project start from tissue culture lab. Thank you.
Thank you, Mr. Zhao. Very
Yes.
excellent presentation. And I can see from the comment from the audience that they enjoyed it very well. Just now, like the name of your company, your work, everything looks very grand. Grand Biotech. Thank you so much.
Thank you. Thank you, everyone.
So now we'll have the presentation, much awaited presentation from all the audience. We'll have this from Mr. Kishore Rajahans. He is very known person in the area of plant sculpture, especially the ornamental plant sculpture commercialization. He is my very good friend, and for last 2 decades of my association with this industry, the commercial industry, not for the research. Initial part was research, but 2 decades of association, I have seen him and his organization growing very well. So thank you, Mr. Kishore, for joining us today. And Mr. Kishore is the director of Care Bioplant. This company is the largest plant sculpture company located in Pune, working in the ornamental. Pune is just 150 kilometers away from Mumbai, and a very nice place. This laboratory is located in the campus of 52-acre, very huge facility, 4 units, working on many species like Gerbera, Ammonium, Phalaenopsis, Dendrobium, Lily, Carnation, many ornamental plant species. And the 75% plants are going for the export purpose. And this company, Care Bioplant, they have joint venture with 13 other companies worldwide. So you can see the level of operation. More than 1,200 people that are working in this organization. And it is very excellent facility in India. So will be benefited by presentation by Mr. Kishore. Mr. Kishore, or to you.
Thank you. Respected dignitaries from BCIL and APARI, respected scientists from ICR, IIHR, and other countries from Taiwan and Thailand, respected industry friends and the farmers from India as well as overseas. Expand your vision and do not be afraid to set the goals which scares you. This is the motto at Care Bioplants for last 30 years. We started this company in 1992. This year, we are celebrating 30 years of excellence, and still we are marching toward the new pinnacles of success in the future. We started this company with 8 people and 25,000 plants first year. Today, we are over 1,500, and we are producing about 80 million plus plants per annum and exporting these plants to almost 35 countries around the world. To be sustainable for 30 years, there are 3 key points: great vision, which was provided by our promoters, great infrastructure, and financial management. That is the most important thing in any business to sustain. And last but not the least, the people, the passionate people. So Care Bioplant is a great recipe of these 3 ingredients or 3 spices. I will share my presentation. Can you see the presentation?
Yes, yes, it is there. Please begin slide so much.
Okay. K Bioplants is a joint venture between Kumar Properties from India, and earlier it was Florist Holland. Today it is Hilvada Florist. So this is a very much successful Indo-Dutch association over last 27 years. This company is in Pune, which is about 150 kilometers from Mumbai. The facility is spread over 20 hectares. We have a state-of-art facility. We have 4 laboratories, each about 3,000 meters square, producing between 18 to 20 million plants per laboratory. This year, we are putting 5th laboratory, which will be in functional by November 1st week. So by end of this year, K Bioplant will produce over 100 million plants. And I think by any scale, this is the largest production center in the country. Predominantly, we are producing ornamental flowering species. Of course, Gerbera is our unique product, USP product, butter and bread. But apart from Gerbera, we are producing Phalaenopsis, we are producing Calla lily, Echinacea, Rudbeckia, Limonium. Then we are producing Geranium, Gypsophila, Dendrobium, lilies, and a lot of other products. We are working on about 19 various ornamental species for India as well as overseas. As Dr. Shukla said, we have a collaboration with almost 13 different companies, but we have 2 major joint venture, which I will say equity partnerships. One is with Hilvada Florist, another with Floricultura. And apart from this, we are working with a lot of other, let's say, eminent breeders worldwide to produce their genetics at K Bioplants and export them either ex-agar plants or hardened plants. Futura Bioplant we started in 2008. This is a joint venture between K Bioplants and Floricultura from Holland. Today we have 2 dedicated plant tissue culture facilities for Floricultura. We produce about 43 million plants to 45 million plants per year, which is for 100% export for Floricultura's market worldwide. K Bioplant supplies 60 million and let's say 43 plants from Futura, so about 100 million plants per annum to almost 30 countries around the world. Our market share globally for Gerbera is about 78%. Every single plant produced for Gerbera and supplied by Hilvada Florist is from K Bioplants. So we take pride as a country and as a company that we have a, let's say, we have put dent in the universe for Gerbera. As far as Indian market is concerned, our market shares are about over 90%. We cater to almost 6,500 growers all over the country. And this key to success is the technical support. We have a team of almost 22 people who are masters agriculture, horticulture, plant pathology. Their task is to give online technical assistance, physical technical assistance through visits to all the growers where we supply the plants. And this technical assistance starts right from site selection until to help him to sell his flowers to reliable buyers in the market. And this has a structure. We have a written report which has been signed by the farmer and signed by our executive. So we follow up almost 6,500 growers year round for last 25 years. This slide is especially on the request of Dr. Shukla. This is just indicative balance sheet or the, let's say, investment returns and the cost. This is, I have taken from the latest March financial balance sheet. So any high quality infrastructure lab will cost you something like 36 crores. In US million dollar, it is about 4.5 million US dollar investment to set up about 3,200 square meter facility. In this facility, we produce about 20 million plants. I have calculated average price, which is about 17 Indian rupees. That is about $22 cents per plant. So for 20 million plants with $22 price, our expected turnover per year is about 4.3 million dollars. In Indian rupees, it's about 34 crores. Now, if you go to the cost component, the cost of production is about 55% of the turnover, which is about 2.3 million $. Administration and selling overhead cost is about 10%, which is about 0.43 million $. Financial cost, since we are a joint venture with Dutch company, we are getting international loan facility. So the interest rate is about 5%. So the finance cost is about 0.23 million $. Depreciation, we take about 10% of the investment. So it's about, let's say, 3.6 crore or 0.45 million $. Now, net profit is about 6.5 crores, which is a decent as far as the tissue culture industry is concerned, and to sustain the facility, to sustain the industry. So which is about 0.8 million $. So if you take the percentage, the net profit is about 19% of the expected turnover. Now, if you add your depreciation into net profit, that gives you cash net profit, which is about 10 crores. So that is about 1.26 million $. Per annum. So the percentage of cash net profit is about 29 to 30%. And if you calculate returns on investment, that is net profit versus capital cost, then it is about 18%. So with these inputs, the cost and the returns, I think the payback period is about 3.5 years. I am presenting this slide for the new entrepreneurs who want to come into tissue culture, that yes, this is not lucrative, but this is certainly decent profits, and you have huge potential to come into this field. I would like to present this slide specifically for the policymakers like Dr. Patole, Dr. Sharma from BCIL, and the other dignitaries. Today, Indian tissue culture industry is really in a stage where the whole world is looking at India for supply of quality planting material, because I think there are two reasons. One, labor issues and the energy issues. The whole Western world today is facing the crisis, either the availability or the cost. So on this scenario, we have a huge potential to produce tissue culture plants and supply to these countries. However, in the last 7 years, we are facing an issue of PRA, that is called Pest Risk Analysis. This is becoming like a stumbling block to the global plant tissue culture business and the opportunity for the India. So my request, humble request to the policymakers, that can we abolish this PRA procedure, at least for import of in vitro planting material. Why in vitro planting material? In vitro planting material is scientifically and physiologically sterile, and if tested negative for various pathogens and viruses, should be no threat to flora and fauna of any country. Also, since this is purely imported for export production back to the source, there is no harm to the local ecosystem. In this scenario, it is my request for the Indian policymakers to look into this issue to waive off the PRA, at least for in vitro import of planting material. Air Bioplants today is a successful Indo-Dutch business model, and we take pride for partnering in success of Indian as well as global floriculture and horticulture industry. I once again thanks to my friend and honorary dignitary Dr. Shukla, Dr. Sharma, Apari, and all the dignitaries and my grower friends. Thank you so much.
Thank you, Mr. Kishore, for this wonderful presentation, and also giving that insight about the investment and the return part. Because this is very important for any entrepreneur, those who are willing to set up any commercial laboratory. So thank you once again for joining this webinar. So with this, we'll have next and the final round of poll with the audience, because we wanted to make it very interactive, and all feedback insights are very important. So I request my colleague to post the last and final poll to the audience. So there will be 4 questions. First, do you agree that micro propagation is an efficient technology and profitable for the production of ornamental plant at large scale, as Mr. Kishore has already presented on this aspect? Second one, about the webinar, how do you rate this webinar in terms of enhancing your knowledge with reference to technique for production of tissue culture of ornamental plant? Third, about the application, would you integrate knowledge gained during the webinar in your research and extension program? Are you going to apply it for your further work? That is the question. Now, fourth one, does this webinar series encourage you to popularize tissue culture technique and establish a new tissue culture facility in your country? So 4 questions. Already it is on your screen. Please complete it in few seconds. The last 5 seconds we'll close this poll and we'll announce the result. Yes. 3, 2, 1. Okay. So result is there. You can see that 99% saying that it is efficient technology and a profitable thing. They agree with. Now this, how do you think that this is a webinar is in terms of popularizing the quality plant, how it is enhancing your knowledge? 64, they're saying excellent, 28 good, 7% good. 1%, they're saying there's still it has the scope of improvement, fair, just 1%. Majority they're saying excellent or very good. Now 95% they're saying that they are going to integrate this knowledge for their research and extension program. Very nice. And that will be the real outcome of this kind of webinar. And last one, 95% they responded that this is popularizing that tissue culture concept, and definitely they would promote or they would like to undertake establishing new setup in their country. So thank you for participating in this poll. Please unshare that result. So with this thing now we have completed all the technical session, and now yes, one thing I missed to so that there is one video of that cave bioplant. So we'll like to project it from our side. It's a very interesting video, and this kind of video is appreciated by all the stakeholder participants, and especially young entrepreneurs. They get idea how this process takes place in commercial level. I request my colleague to project that video.
An ultra-modern laboratory spread over 12,000 square meters. State-of-the-art temperature-controlled greenhouses stretching over 6.5 hectares. 750 highly trained technicians monitoring the delicate multi-stage process. Spacious growth rooms capable of storing up to 20 million plants at any time. Exports exceeding 25 million plants per annum.
Continuous innovation and consistent quality are the cornerstones of our success story. Over the past 2 decades, we've established a benchmark in the plant tissue culture industry in India. I am happy to say that we have succeeded in expanding the boundaries of the floriculture business vertically and horizontally in the country and abroad. Our collaboration with global giants has paid us rich dividends.
At the heart of the entire process lies the ultra-modern lab facility at Pune. Nurturing in vitro plants demands the highest levels of attention. Cloning of plants is a very delicate process. At KF Bioplants, it receives the utmost care. Built-in safeguards ensure that the plants receive a 100% sterile environment to prevent infection. Once placed in the media, each plant is carefully preserved for the required period of incubation. Plants enter the temperature-controlled greenhouses fully protected and leave fully acclimatized. A state-of-the-art fertigation system provides steady supply of quality media. A fully computerized ERP system ensures faultless precision. Precise scheduling and excellent logistics make sure that dispatch of material to numerous destinations is both safe and timely. At KF Bioplants, improving facilities and processes is part of the company's DNA. Upgrading from autoclave to N-Biojet, using microwave energy to ensure faster media sterilization. Installing LED lights in the laboratory to ensure energy conservation. Bringing in somatic embryogenesis for rapid mass propagation of plant. Commitment to quality and consistency has earned the company ISO certification and DBT accreditation. KF Bioplants, India's largest plant biotech company, producing 60 million plants per annum, handling every stage of the business, offering technical assistance 24/7 to farmers and growers all over India, collaborating with the global best for the latest technologies, processes and products.
Thank you. So this kind of celebration for the ornamental presentation. We have seen the presentations, video, and a nice interaction with all the audience. So now we are coming to the final session. That is the open question or answer session, open question answer session, and the concluding remark. For that, I would invite the very important person for this webinar and instrumental for conducting such webinar series, Dr. Rishi Kumar Tyagi. Dr. Rishi Kumar Tyagi is working as a coordinator of the FCOP in Apari, and he is PhD in botany from Delhi University, India. And he has served 40 years in ICR system, and he was heading before Apari. He was heading the division of Genoplops conservation of ICR and NBPGR. He has published a number of research papers, more than 100, authored many books, joined a very invited lecture, keynote presentations, and presently he is promoting agri-biotechnology, a global platform, conservation of bioresources, sustainable development at a Apari platform. So under his guidance, we are organizing such important program. So Dr. Tyagi, over to you. You please kindly moderate the question answer session, and also place that concluding remark once question answer is over. Over to you, Dr. Tyagi.
Thank you, Dr. Shukla. And before I go for the question answer, I also join others to wish you a very happy birthday. And you have rightly mentioned that you have got so many orchids at the present from different part of the country. So I think you must be very pleased, and Mrs. Shukla also very pleased to celebrate on this screen by having the bouquets of flower from the different countries.
Thank you
So
much.
I come to the question answer session, which is very important, and Dr. Shukla is kind enough to allocate this time separately, because generally in webinars, the question answer time is not left out, but we give the importance and we attach very importance to question answer session. And we have got a lot of questions. This shows that the generation of the interest by the participants in this webinar series as a whole, and in particular this ornamental crops also. So I may not be able to take each and every small or major questions, but certainly I will pick up the questions which are very relevant to today's discussion and presentation. So I'll go through one by one, and maybe I'll request the expert to my understanding to answer the question. But if somebody else wants to supplement the answer, they are also welcome. There is one question is that probably Dr. Ashwath or Dr. Kanchit may look into that. Some Madhubala is asking that which media and explant is best for orchid tissue culture. Can you, Dr. Ashwath and Dr. Kanchit can answer this?
I think Dr. Kanchit is better.
Oh, okay. Maybe I start first. So for orchid, mostly we use a vaccine and vent and modify with banana or coconut water. And if it doesn't work, maybe try the MS medium, half MS medium, Knudsen C, something like that. I think that's enough.
I would request Ms. Madhubala that she may contact Kanchit via email for the detailed discussion on that. And it was mentioned earlier that this webinar is being attended by several countries from Asia and Africa, and I find I think the participants whose I'm going to name is from Africa. Nathaniel Sharif is asking that what is the temperature and light intensity needed for greenhouse growth chamber? Dr. Ashwath or?
Yeah, see, greenhouses normally for a good hardening, around 30 to 35 degrees centigrade is the best temperature. Humidity, if it is more, it is good. Above 80% is always good, 80 to 85%. And especially in the initial time, it should be more. It should be more than 90%. And then in the secondary hardening, you can slowly come down also. Temperatures up to 35, easily you can go, there is no problem. But if you have a good humidity. See, one important thing which I see is the light. See, light normally we requires around 50% of the light, we say, but it depends on, what type of, how much light is. Normally, if you say by a lux, around 1 lakh lux will be there outside. So we give around 40,000 lux to 30 to 40,000 lux is required for a hardening. So that is where you have to be very careful, because many times there will be a cloudy days, as Kishore sir said, they have a sort of reclinable shade nets will be there. But many of the polyhouses, they just put a shade net, 50% shade net. And during cloudy days and during rainy days, light seepage is very, very less, because already 25% light will be cut by the polythene sheet. So you should be very careful with the light. So light is very important factor for even development of the roots. And watering, again, water is a very good water is required for a hardening. Salty water above TDS more than 600 is not really recommended. And if you have a rainwater, that is the best source of water for the polyhouses.
Thank you, Dr. Ashwath. Another question is probably it is for Dr. Kanchit. That question is from Reniel Pamplona. That I would like to ask, how much do you usually sell the orchids internationally? Is it in bulk order or per piece? Dr. Kanchit, I think question was raised during a presentation from Thailand point of view.
Yeah, I think depend on the seasons. During winter may be more expensive because we have less production. And usually we sell by spray and the market price and from the farm price are a lot different. From the farm price, maybe 5 baht, 6 baht. But to the international market, maybe $1, $2, something like that.
Thank you, Dr. Kanchit. And Dr. Shukla, there is one question raised by many participants that about the training program of the ORCID. So I cannot make out that country origin. Some are from India, some are from other side. So can you throw some light about this question?
In fact, for the training program, BCIL is considering one initiative to include different module, different crop, and a different level of training program. So I will suggest them to keep on visiting BCIL site, and we have noted their comments. And whenever it is started, we'll write back to all the participants those like this question. So we'll try to organize this program in association with industry so that they will get exposure of R&D plus industry, and definitely we'll take help of Dr. Aswat also how to formulate this training program.
Yeah. Okay.
Thank you, Dr. Shukla. Another question is from Nang, that how many multiplication should be done, or any segregation or inbreeding failure can cause after several subcultures. I think question is that how many cycles of subculturing should be done so that the segregation or some other problems are not occurred. Dr. Ashwat?
Yeah, see, it depends on the crops again. See, for ornamental crops, not really, like they are not very stringent compared to banana, where banana normally around 8 to 10 subcultures will be there. In Gerbera, one of my PhD student, she studied. We have even up to 22 subcultures. We did not find much of any variation or even loss of vigor. So it can go up to like 15, 20 subcultures. It's no problem. But as like industry, if it gives lesser the subcultures, it is the vigor of the plant is better always. So don't go for too many subcultures. It is not really fixed. The research has not really says that this many subcultures. But as such, we can definitely see where in South Korea, I have seen in orchids, as the number of subcultures increase, the number of plants which are thrown out will be more actually. When they take out the bottle, when the subculture is around 12th culture, it will be around only 10% of the plants they use, non-vigored plants they used to throw out. When it comes to around 15 or 16 subculture, the number of plants will increase. So definitely a person who is really got a good knowledge, when they open, they can say that, what is the vigor of the plant? Because if the vigor of the plant, looking at the vigor of the plant, we can stop the subculture. So they can contact me for any further details, and I can put them in a proper.
Thank you. Thank you, Dr. Shukla. Again, a question directed towards BCIL. Very excellent and useful software you have launched today. And several questions are that how to access that software. Is it in open access or it is available for the general public or for the BCIL only?
In fact, just a few seconds, I'd like to tell the background. For the certification program of Government of India, what BCIL has played key role for establishment since its concept till commissioning and operationalization last year. When we visited all the commercial setup, all the companies, majority of the companies, more than 90%, they are facing difficulty in maintaining their records and documents in organized format. And also, when we talk about the traceback system, from the dispatch of plant till the mother plant, there is quite difficulty in organizing those records. And when we ask for certain record like contamination, performance, they're all running around the different section to get those records to document. In this software, very small piece of application which integrate all the operation, so that not only for the record keeping, this also give idea of the production schedule. Suppose in the morning, how much media I have, how much culture I need to transfer. or how many plant I need to send it. So this will help. Now coming to the question, as I told that nowadays government, they are focusing on the self-sustaining program. So this program has been developed, is a licensed program, it is registered under the trademark, and will provide to the companies on a cost basis. I will write to the company, I will inform the company of the costing, but cost will not be huge, it's a reasonable cost, but it will be on a license basis. And once they will avail this software, will provide all the training and the maintenance part, hand holding for the operationalization. That I write to all the participants, those who raise this question, or they can write to me. I will provide all the details.
Okay, thank you. Thank you.
Thank you.
One question or 2-3 questions related to this is that what is the cost of the bioreactor? I think Dr. Ashwath, you gave the idea of the small scale bioreactor and the big bioreactors.
No, I don't know much about the big bioreactors. Whatever the bioreactors which we are using in lab, we got it assembled. There is one company in Bangalore, in Vascote. I can give their address. One Dr. Ravindranath. He can make bioreactor up to 50 liters. He can make. So the 5 liters bioreactor with one small 5 liters bioreactor, which costs around 4,000 to 5,000 rupees. So if you can have more than 10 bioreactors attached to one pump, so which costs like 5 bioreactors, 5 liters bioreactor, about 10 with a pump will cost you about 1 lakh rupees. I can give that address. Somebody contacts, I can give their address. But I do not know about the bigger bioreactors because so far I have not seen 2 lakh. In fact, I have seen in South Korea even up to 50,000 liter bioreactor, which I am not really aware of.
Okay, thank you. And another one question to you that you mentioned in your presentation that to avoid the vitrification, the cooling of the bottom of the.
Yeah.
So how to do it?
Normally,
is asking how to do it.
Dr. Kishore has shown they use this LED lamps. Whenever the LED lamps are there, that bottom cooling is not required because it will not produce any heat. So the traditional lamps, they will have like a non-LED lamps where they produce a lot of heat, under when the bottle is kept on the surface. So what you have to do is you have to put up the lamp, like light during night, so that the media temperature and the top temperature should be only around 2 degrees should be there. So normally if you use LED lamps, this problem of bottom cooling will not come. So it is only, otherwise you have to keep it on a net where good aeration will be there. So that time also there will be no problem of your media should not get heated up because of the light and the temperature. That is the simple thing actually, which I have seen in, of course, way back in 2020 in South Korea. Most of the labs now they keep on the net. Probably you have seen even at Kanchit, the bottles are kept on the net, not on the hard surface where the heat is produced. So that is the thing, a simple thing.
So where from the genuine gerbera and orchid mother plants can be obtained for tissue culture? Dr. Mallikarjun is asking for.
In fact, nobody will give the mother cultures because that is the trade secret, like mother cultures. And we are not supposed to multiply the KF varieties. Of course, in IHR now we have released 4 or 5 varieties. If they want, they can. They have to buy the planting material. They have to take a license from IHR about the variety as well as the production. Once if they buy licensing, then we give the 25 tissue culture bottles along with the even the media constitution, everything we give. They can contact me. We normally charge only just 50,000 rupees for a variety, so that we give the license for production as well as complete protocol of production of those planting material. They can contact me anytime.
Thank you, Dr. Ashwath. Mr. Jha, a question for you. Do you provide the training to the students also? Dr. Suchmita Shukla is asking this question. Mr. Jha.
Yes, and I am curious, what is your question for production? What plant? I cannot really understand.
Question is that do your company provide the training to students also, university students?
Yes, we business for both American and also Netherlands and also Japan. And everywhere we ship to our plant. Does this answer your question?
Yes, yes. Thank you.
Thank you.
I think I had taken up almost all the relevant questions. Just give me a second. I go through. If something is important is left out.
Can I have one question to Kishore? Dr.
Yeah,
Rajan?
please. Yeah, please.
Sir, in fact, we have a PhD program. AIHR has got a PhD program in flower crops. See, some of our students want to have a sort of industrial, visit, or even for a sort of, like now under NEP, even we had to give them 3 months or 6 months placement in the industry. So is it possible, Dr. Kishore, to have a sort of industry, visit or, industry placements for 6 months for our MSc or PhD students? Is it possible in your company?
Yeah, Dr. Ashwath, basically industrial visits are very much possible because we get a lot of students from different universities throughout the country from abroad. So yes, please, you can organize that and we'll take good care of them and show them around. As far as the training program for longer period is concerned, will not be practically feasible. But let's say a training program for a week or let's say 10 days, yes, we can pretty much organize that. And we can give them the focus activity as per their requirement of the study. So that is possible.
No, this is requirement of 3 months or 6 months placement. That is now under NEP. For any student to come out of MSc or PhD, they have to have an industry placement for 3 months and 6 months. Of course, we send them to a small industries, but I found that, at least as a giant, you can give a lot of human resource management to our students so that they will also become, future industrialists like you. That's
so
what is my
for
intention.
3 months, yes, definitely we can do that, sir. Absolutely no.
Okay, we'll write to through our director.
Sure. That will be pleasure for us. So no worries.
Thank you.
Thank you, Mr. Kishore. Dr. Shukla, question for you. Is there any facility to test the ornamental disease-free plant material in India?
Yeah, IIHR is doing this excellent job. Dr. M.K. Reddy is there.
Yeah, now IHR, we have, our lab has been, of course, pathology, Department of Pathology has been identified for a quarantine and to give a certificate. We take up all the ornamental plants. There is no problem. They can contact. We have a small fee for that. Of course, depending on that, we can get some, give the certificate.
Yes.
One question is how we get our e-certificate. Adnan Riaz is asking, so how we get the e-certificate?
E-certificate, those participants attended actively and participated at least in one poll will provide the certificate. In fact, all the participants, those are getting certificate, they're proudly sharing their social media also. I could see that. So it's very good that it is adding value to their knowledge. We'll provide the certificate to participants.
Okay, so I think I have covered major questions and some questions are left out which are very general nature and rather scientific nature, which require more discussion and more information. So participants may request the experts to further contact for their clarification. But one issue was raised by Mr. Kishore Rajhans about the PRA. And I saw in the question answer session, one of my old colleagues in NBPGR, Dr. Celia is there. So I think I would refer Mr. Kishore to Celia Chalam. Dr. Celia Chalam is the head of the quarantine division in NBPGR.
I'll connect you.
You can please. Contact Dr. Shukla can give. And she just want to understand what is the issue and how she can help you in that way. So thank you, Celia, for if you are here. Thank you, Celia, for your
thank
intervention.
Thank you.
But I had raised one, Dr. Shukla, about Kishore. Can you just respond, sir? Multiplication of different ornamental plants now. A small person definitely is not possible to start with. I say if you want to start with a caladium, if you want to start with a dracaena, it takes a lot of time for. So, is there some sort of some sort of service which one can do within the local? See, of course, for export, then a lot of problems may come. For local plant production, whether we can do that type of service, whether it is ICR or along with the industry, to start to industry, starting cultures. That is one thing. In fact, when last time when Dr. H.P. Singh was DDG, many of the industries, they told us that ICR, IHR should supply the planting material, initial planting bottles. I mean, especially for banana, for G1 cultures or G2 cultures, but it did not take up. But this is the time, I think, BCIL or DBT, somewhere we have to start, so that one starting cultures lab, if it is there, then we can definitely help the lot of starting companies. That is what I feel, Kishore.
Just to supplement and inform the audience that BCL has already initiated mother stock culture production initiative.
Excellent.
So that we started with the banana, because largely company they are doing banana.
Yeah.
And that we had done some way testing our concept. So selection of plant till the providing culture, that is virus tested. But definitely, if BCL is doing this thing, we have a bigger plan. And with the help of industry leader like Dr. Kishore Rajhans, definitely we'll try to include all the crop in commercialized crop in near future. So that is our plan. We'll do it. It is a difficult task, because for last 20 years we have been discussing this thing different forums. But I think if BCL is engaged, then with the help of industry partner, we can do it. Under core government research institution, it will not be feasible because their mandate is different. But with the help of industry, some kind of public partnership program, we can start in a self-sustaining mode. Banana already we have done. One batch we supplied. In coming February, March, again, we are going to provide the next batch. But definitely we'll keep on adding new crop on that.
Thank you.
Thank you, sir. Thank you.
So thank you, experts, and thank you, participants. I hand it over to Dr. Shukla. The question and answer session is over now.
Sir, now the final session, that concluding remark of the final event of the final session, it is over to you.
Oh, okay. Thank you. Thank you so much. All the esteemed experts and the participants from different countries. I'm told that participants are coming from 45 countries from Asia and Africa. So it is quite heartening and satisfying at the same time that we are concluding this webinar series, which was started in May 2022 in collaboration with BCIL. And in fact, when we started talking about that, how we had to arrange it, Dr. Shukla worked it out very meticulously that we have the 4 major crop groups where the tissue culture is applied very prominently and very profitably. So the component what we had, very good blend of the academia as well as the industry. In all the 4 webinars, we had the experts from the academia side, which are giving the information on the protocols and all the different crops. But the experience from the industry is a unique one, which have been included in these webinar series. I'll not go into detail of the others webinars, but today webinar on ornamental crops is very, very, in fact, what Dr. Shukla, I borrow your words, that it is a treat to all of us. It is a treat to all of us for the knowledge, and it is a treat to all of us to the eye-soothing to see different kind of the flora, these ornamental crops and the flowers. And again, very happy birthday to you, Dr. Shukla, on this occasion. I heard all the presentations, and they are really very, very excellent presentation. Dr. Kanchit mentioned about the low-cost technology, how in Thailand they have developed low cost. And for the startup people, that low-cost technology will be very useful. And of course, one can scale it up to the level of the grand biotechnology or the KF bioplants level. But when the purpose we are looking for this webinar to attract the youngsters, young students or the young women to come into the business of the tissue culture, then the low-cost technology is very useful, what Dr. Kanchit has also discussed. Then Dr. Ashwath has covered several crops, and I think I enjoyed, Dr. Ashwath, your presentation, though I have been working in tissue culture for a long time, but I have worked on the ornamental crops. So ornamental crops working on tissue culture is two-way good. That one, it is your profession. Another is that you always feel very happy mood by seeing all the flowers and visiting the greenhouses and all that. So you have mentioned two important points, and I think two industrialists are there, two industry persons are there. One is that that the big companies should have the support to the smaller company. And see, the Asia or be it Africa or in Pacific region, that we have the smallholder farmers. And the new startups are coming up, but unless they are getting some support, I'm not saying the trade secret support has to be shared, but certainly some logistic support, some capacity building support can be provided to the smaller company or startups, so that, yeah, I can understand that it can generate the competitive, but I think for the humanity's sake or for the country's sake, this much is required or expected, as Dr. Ashwath has mentioned in his presentation. Another important thing, what he has mentioned, that yes, I think India is coming up gradually, or some other countries in South Asia, but in East Asia like Taiwan or the Thailand, Thailand is having very excellent infrastructure. The private sector is supporting the academics to develop the protocols. They are supporting, they are having that what is their need to develop any research, and they support, and this research is utilized by the industry. So Thailand was one example. Taiwan is another example where so much technology has been developed in the small country. And I just want to show you one thing, that Taiwan has a developed technology. I don't know what it is called. I was shared by one of my friends, the chairman of ATRI, Agriculture Technology Research Institute. If you can see this, I don't know, you are not able to see because of the background.
See.
So what they have developed, a plant, a orchid, showing the blue and yellow leaves, half of the blue and half of the yellow, and to support the Ukraine war, to support the Ukraine during the war. And so they have Mr. Jha must be knowing what is this technology. The leaf of the petals of the half are blue and half are yellow, the color of the Ukraine. So I think wonderfully what Taiwan they have done in the public and private partnership is excellent example. So Thailand, Taiwan, both are the good example. So Dr. Ashwath, I think I fully agree with you that I don't know that whether the dialogue, this public-private partnership we are listening from since our birth in the ICR or in the research, but it has to go in a long way. Dr. Jha, your presentation was very excellent, and it was having the emotional touch and the current situation. We have the emotions always for the conflicts between the China and Taiwan, which you have also reflected in your presentation that why Taiwan matters, and Taiwan matters for the world a lot. And I don't go into the other electronic industry and all that, but certainly in the floriculture or the ornamental crops, what the export potential you have shown from this country, it is a very good example. And Mr. Kishore and Mr. Jha, this platform also gives the opportunity to provide the platform to the private sector also. It is not only the public and private sector is the important partnership is important, but public private partnership is also important to foster your business, and how these synergies can be developed between private and private partners. So you know, and I think for other crops also, Dr. Shukla has the experts addresses, and you can come in contact with them.
Sure, thank you.
And
Sure,
I
thank
think
you.
the presentation by Mr. Kishore Rajhans is very encouraging and very eye-opening, in fact, that 52 acres, the management and all that. And Dr. Shukla was telling me off the screen that this is one of the largest company for the, and you have shown that what is the potential of the export in that way. You have also touched upon the economics, of course, of the bigger state, but certainly the economics is important for the new agripreneurs. And thank you for acceding to our request. We made your request before yesterday.
Thank you.
PRA, I have already mentioned, you just contacted Celia, how she can help and how it can, what are the issues in that way.
Yes, sir.
I cannot say that I am extremely satisfied with this webinar series as a whole. And this ornamental webinar today, what we have. The purpose of this webinar series was to create the awareness about the importance of the tissue culture, which is one of the important part of the biotechnology, which can be very profitably used by the smallholder farmers, for the smallholder farmers or the young agripreneurs to improve their income or to improve their livelihood. The another purpose of this webinar series is that what the poll questions have been dropped every time in the webinars, that how to understand the pulse of the different participants of different countries, that how tissue culture is important and how tissue culture is required in the different countries. So this information will be very useful, and we have a plan because the capacity building is invariably have been asked by the participants to have a program. So we are looking forward for some funding agencies or these so donor agency to develop a big program for the capacity building in the Asia Pacific region. So I hope that with the experts, what we have listened, and with the expertise which is available in the Asia and this region, we will be able to develop a good program. And maybe the experts will be contacting later stage also, that how again you can be useful for conducting those programs in case we develop any program at the regional level. I will not take much time now because it is already exceeding the time, but finally I would like to convey my sincere thanks to BCIL for being partner with us, particularly Dr. Purnima Sharma, the Managing Director, Dr. Shukla and his team for working out for all this series, developing the agenda and so many things, and handling the webinar logistics and the background. Dr. Shukla and your team, I'm very thankful to you. And I'm thankful to Dr. Ravi Khetrapal, the Executive Secretary of APARI, my esteemed colleague for a very long time, and now in APARI, with whose cooperation and guidance we have conducted this webinar series on the demand of the different countries in the Asia Pacific region, which was collected, this information was collected through a survey earlier. I am extremely thankful to the experts today who have made the excellent presentation. Professor Kanchit. Professor Kanchit, I met him in Thailand, in Bangkok. He is a very renowned person in the University of Mahidol. And he has produced a lot of his students. And he has a big battery of these students working on the ornamental crops. I have seen how these orchids, greenhouses are there in the field, or farmers' field are grown in the Thailand, and they are exporting. And the farmers are getting benefit of this technology, tissue culture technology. And I am very thankful to Dr. Ashwath, my old colleagues from ICR, and in fact, Dr. Ashwath and myself, we started career in ICR in research at the same time. So Dr. Ashwath, apart from this academic relationship or the associationship, I'm very thankful to you that after a long time I saw you, your face on the screen. I'm extremely thankful to both the extreme person for the head of their organization to take out the time, Mr. Jha from Taiwan, and Mr. Kishore Rajhans from India, Pune. Both have made the excellent presentation. I am sure the participant must have been benefited and encouraged also to come into the business of this ornamental crops. And finally, I want to thank my colleague from APARI, Jack. Jacqueline is there. Thank you, Jack, for working in the background. And I'm extremely thankful to all the participants coming from the different part of the country. And I know the participants from Africa, they are there, and from Asia, Pacific region is also there. I myself, if you ask me, quite satisfied by having this webinar concluded today, webinar series concluded today. And we look forward with the great hope that we'll find out some donor to develop the capacity in the region to have the tissue culture in the different crops, depending upon the need of the country. So as Dr. Ravi mentioned that APARI is the member-based countries. So we have about 22 countries of the member of APARI, and we have to serve those countries. And yes, India is there, Taiwan is there, Thailand is there. So many other countries are there. So with the cooperation and with the partnership with BCIL, we look forward for a good program with the cooperation of some donor agency so that the capacity building can be further strengthened in tissue area of tissue culture in the Asia Pacific region. With that, thank you once again to everyone. And I stop here unless Dr. Shukla has a final word. Thank you so much.
Final word, I have to say thank you to one and all. And always we exceeded timeline of 1 hour, whatever we decided. In spite of that, there are consistent participants there. So that shows that their interest in the topic and the theme and the presentations. So I think we can close it now. Thank you so much, one and all, to all the experts and the participants.
Thank you. Thank you so much.
Thank you. Thank you so much.
Bye-bye.
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 Bye.
Bye-bye.
Bye.
Bye.